Good morning. This is day 2 of our Nano to Cosmic Studies of Complex Systems workshop. I trust you can all see the kind of filler slide of the Earth and Europe as we get started. So for today, we'll run through just a couple of quick housekeeping measures. Our purpose today is we're going to shift gears a bit. Yesterday, we surveyed, how complex or how complicated things can get on the early Earth and how we're using and drawing upon, diverse data sets to try to reconstruct how, just how complicated and complex things can be. And today, our purpose is to survey kind of current theory and methods related to characterizing complex systems. And it's broken into 2 sessions, but it's in some ways actually kind of 2 parts or 2 halves of the same coin, and you'll find some overlap. I just want to point out that yesterday, for example, I think there was a question or comment by Stu Bartlett to, I think it was Wilhelm Huck, if I remember correctly, something about whether the chemical system that he was studying was capable of Hebbian learning. And I was actually pretty excited to see that question come up, because I think if yesterday's sessions did anything for you, it's to show how close we are in terms of going from systems chemistry to all of these realizable examples where the network is capable of doing more than just what we can reduce to, say, physics or chemical descriptions. It becomes something broader, and we need a different tool set to analyze and to begin to make sense of, what a systems level behavior might be when we get many different components with different levels of recursive feedback or input or output. And so that's really what our purpose is today. For meeting logistics, just like yesterday, if you need any tech support, reach out to Mike at any time. If you see any instances of harassment or abuse of either the Zoom room or the breakout rooms, contact Donald Burke Aguero. For today, the session is again structured much like yesterday. We'll have 2 3-hour sessions separated by a break in between. Our purpose is to talk about unifying our views of origins research by depicting these kind of disparate experimental approaches as best we can on some kind of unified spectrum or under some kind of umbrella of being able to compare when someone is working on, say, polymerization chemistry like Nita Sahai, or if someone's working on, highly ionizing atmospheric chemistry like Vladimir Arapetyan, or any number of people who study the icy outer moons. Is there any way we can kind of sensibly compare and contrast and put all of these activities on a somewhat equal footing and realize that no matter what part of chemistry or the environment you're looking at, it's all connected and must be connected in terms of the ability or the tendency to produce entropy. That's kind of a very broad theme for today. And why is a unified view important? I'm going to blast through this very quickly because you're going to see this and we're going to discuss this in the days to come. But as I outlined yesterday, NASA studies the largest complex system of all, the universe. And it really draws on information from all parts of the electromagnetic spectrum to try to piece together what they're looking at and how it functions. And as we look out across large scales of space and time, starting at the largest scale of these kind of supragalactic distributions of mass and energy, we see a recurring theme as we go down to the structure of the distribution of cosmic rays, the energy outputs of stars, and all the way down to the planetary scale, where our energy distribution of our planet is represented or in one way by the magnitudes of earthquakes and the attenuation of internal heat energy and dissipation to the heat bath of the universe, and all the way down through biologic structures. You see this recurring theme of connections and distributions of energy on these kind of log plots. Now, it's one thing to measure these things and to note that that's what's going on, but it's quite another to try to disentangle what the mechanisms of energy transfer are and whether they're significant. These scaling relationships are implicated with the potentials for these very interesting attributes like nonlinear responses or hierarchy or effect propagation that goes beyond just diffusion. So, either encoding information or having some attribute of the system that is heritable over time. all of these are implicated, but it's not necessarily automatic that they follow in all instances. So there is some nuance there to understanding whether we're dealing with a system that is just I go back to these terms, very complicated, involves a lot of parts, and a lot of those parts may be interacting with each other, or genuinely complex and defying kind of the tendency of systems to relax the equilibrium or just to rely on diffusive processes to dominate the flow of matter and energy in a system. So our present theory, the reason this is relevant to prebiotic chemistry is that, we know that life manifests a lot of these attributes of complex systems, and we would suspect that prebiotic chemical systems also manifested some degree of these attributes. We just don't know how far back or to what full extent, and even if we suspected they were there, how would we be able to measure them or disentangle them from some other explanation in the system? It seems easy enough to try to look for these top level attributes, but it's quite another to, again, go back into our laboratory experiments, many of which were just summarized yesterday. And I hope you got a sense that we definitely feel, it seems like we're at the cusp of something. We have numerous example systems, whether it's the amyloids, polymerization systems, RNA interactions. We have now a small array of systems that genuinely seem capable of exhibiting complex behavior. And these might seem like little keyholes into a broader array or patterning of ways in which organic chemistry can become complex and maybe become like life. But each of these is pointing to the fundamental need for new tool sets and new theories to try to make sense of what's going on and to try to design tractable experiments that will help us advance our knowledge of the origins of life. And there are a lot of really massive unanswered questions when it comes down to try to disentangling what's actually going on in a system and whether we can correlate it with the likelihood or the propensity for generating living systems from scratch. So that's really what our talk is about today, and what the array of talks is about today. So I'm kicking off our first session, which is about network tools, theory and analysis and applications. And we've got a sampling of 4 distinct speakers. Each of them are going to touch on different techniques or different approaches to using and leveraging network analysis tools or network modeling tools, and to try to describe behaviors that might be lifelike. And we're taking a slightly different format. These talks will be a little bit longer in duration than the talks yesterday, mainly because we want to afford you the chance to be introduced to these topics. Some of us may not be as familiar with them as others. So we're trying to give our speakers as much time as possible to unpack their ideas. And with that, it's my great pleasure to open our session with Ginestra Bianconi, who's joining us from the UK. Ginestra, let's see, I'll stop sharing.
Yeah.
Are you there? And are you ready to present?
Yes, I'm here. Yes, let me.
Wonderful.
So thank you so much for the invitation, and sorry so much for the state of my voice. This is not my normal voice, but hopefully I will be able to carry on with that. So it's very exciting to be here at this workshop, and I structured this talk with some introductory slide on network model, and then I will cover recent exciting research on higher order networks. So complex networks describe the interaction between the elements of large complex systems, and so they represent protein interaction in the cell as well as the internet, or for instance, diseasome network in which each disease is a node and diseases are related if there are common mutation. So from the theoretical perspective, complex networks lives if in the white space between regular lattices, a completely random structure. And the thing is that the structure, they encode information. And despite this space is very big, they have been shown to display striking universality, including the small word, scale-free distribution, and the fact that they have displayed communities. So the small word implies that the diameter of the network is small, so that with a few hops, you can go and reach any node of the network. Scale-free network means that the distribution of the degrees, how many links the node has, is broad, meaning that there might be hub in the network that dominates the dynamics. And finally, you have modularity, meaning that the structure, there are regions of the network that are more easily, more strongly connected than that. So there are nodes that are more strongly connected between themselves, between the community than across the community. So models for these networks have been classified into three broad classes. Growing network model, which try to explain the emergence of property, how universality can occur. There are also deterministic model, which can be useful. And then there are maximum entropy model that instead describe a maximally random network with given constraint, with given property. So they don't explain the emergence of the property, but they build maximum entropy ensemble with this property. So in terms of emergence, I've been involved in this parapassing model early on in my career, in which you have this model is inspired by biological evolution, in which each node is assigned an energy or, as you want, a fitness, and the network grows. And when a new node arises, it attaches preferentially to popular nodes, but also to nodes with high fitness, and this fitness take the shape of this Boltzmann factor. And so the network proceeds like that. And interesting, this model displays a phase transition. So as a function of this beta parameter, which tunes how much important is the fitness with respect to the popularity of the node, then you can have a condensation phase. So one node grabbing a finite fraction of all the links. And this transition can be mapped to Bose-Einstein condensation. And interesting enough, if you look at the theory, this is also connected to evolutionary theory, such as the House of Cards of Kingdom model. You can construct also other networks that are growing and that are growing in the opposite direction, like Cayley trees that decide where to go depending on the energy of the node. And this, interesting enough, follow Fermi distribution instead, which might appeal with some physicists, people that are physicists background. Then there are deterministic model, maybe this Apollonian model, in which practically you start with the triangle and then isolate the triangle, you put a node in the middle, and then you form three triangles, and you do that iteratively. And these are actually also scale-free. And then there are maximum entropy models, so models that arise from maximizing the entropy of network ensemble given a set of constraints. And if this constraint is, for example, the degree, you can do microcanonical ensemble and canonical ensemble, in which in one case you fix the degree of each node, in the other case you fix the expected degree of each node. And this ensemble, interesting enough, since you fix a lot of constraints, an extensive number of constraints, are not anymore extensive, so the entropy of this ensemble are not the same. differently from what's happening in statistical mechanics. So these are microcanonical and canonical ensemble. Recently, I also work on grand canonical ensemble, in which the labeling of each node is not particularly important. And what you can do is not fixing the degree sequence, the degree of each node, but fixing the degree distribution so that you can allow the number of nodes to vary, right? So these are kind of mixture model. You first draw the number of nodes from a distribution, then you draw the degree sequence from a given degree distribution, and then you draw the network uniformly from on the network with that degree sequence. So practically the network is given by this mixture model, where each degree of each node is drawn from a given distribution. And for this model, you can also solve kind of inference models. For instance, you have a network with n nodes. You don't know the whole network. You know only part of it. And the question is, how can you infer the number of nodes? And in order to do that, you can use a Bayesian approach, assuming that the true model is this grand canonical model, and infer the latent variables, which are the true number of nodes and the degree of the unsampled node. So this is from the information theory aspect of simple network and the structure mostly. And then regarding the dynamics, summary of some of the most important results is that actually the combinatorial and statistical property have been shown to be very important. For instance, if you have this broad distribution, you have that percolation, the network is always percolating. So it's very robust to random damage. And for instance, also the Ising model is always in the ferromagnetic state, so always ordered state due to the presence of these apps. So recently, there has been a lot of work on generalized network structures, such as multi-layer networks and higher order networks. And I want to tell you a little bit more about the higher order networks. Also, multiplex networks are exciting, but I will not have time to cover everything. So higher order networks characterize interaction between two or more nodes, such as in hypergraph, simplicial complex, or network with triadic interaction. So for instance, in the brain, you might have three regions of the brain, and there might be pairwise correlation between these three regions of the brain, given to three pairwise signals, or you can have three-way correlation indicating an activation that occurs at the same time. Or always taking the example of brain, you have two neurons that have synaptic interaction between them, but the glia might interact with this synaptic interaction. So this is a triadic interaction in which a third node, the glia, affect the interaction between the other two. So it's a higher order interaction. And of course, multi-layer networks are also another way to distinguish between other type of interaction between the nodes. So while in simple network, we have seen that most of this interplay between structure and dynamics has been driven by the realization that the degree distribution and combinatorial and statistical property are important to link structure and dynamics. With higher order network, it is emergent that also simplicial geometry and topology are also playing a role. And here I will take as an example the brain, but these are mostly mathematical results, so I hope it will be useful also. So we have some model, both non-equilibrium growing model of emergent geometry and maximum entropy model for higher order networks. And this can be useful to define dynamics on top of them. And my book is a publicity now. And also, if you want, there is this review article in which we show that actually with higher order networks, the interplay between dynamics and structure is really very much the perspective on this interplay is very much new and open. And in fact, dynamics cannot be just defined on the node of the network, but also on the links, for instance, or on the triangles. And you can have these dynamics, and they are tools to project dynamics of links, triangles, and tetrahedra from one dimension to another dimension. So if you have dynamics on the links, you can project on triangles, or you can project on nodes. So these initial complexes are fully alive with this topological scene. So first of all, I want to show this direction, how simplicial geometry is important to describe the interplay between structure and dynamics in higher order networks. So as an example, we saw in the brain that neurons in 2D and in 3D, they have different synchronization property. So if you grow a neuronal culture on 2D or in 3D scaffold, the synchronization property are different. So the mathematical question is, what is the right notion of dimension of a network that is responsible for this effect? And after we published this more experimental work, we went with some more theoretical work using the Kuramoto model, which is a model in which the phases are associated to the nodes. Each one has a different phase, so it's a heterogeneous model. Each node has its own frequency. And then you couple the phase of nearby nodes, and if they are coupled and you raise the coupling constant, then you find a phase transition in which they start to synchronize. So here we have models in which we can change some parameter, which is the spectral dimension of a network. And this is a property that depends on the spectrum of the Laplacian of the network, and is the dimension perceived by a random walk. You might be aware that the drunken man in 2D and in 3D has different chance to return home. And also in lattices, you can define a spectral dimension, which gives the equivalent effect in the return time. And what we show is that for spectral dimensions smaller or equal than 4, the synchronization cannot be thermodynamically stable. So we test this in a model of this higher order network, which have a tunable spectral dimension, and we see that as a function of the dimension, the synchronization start to stabilize. But as long as this spectral dimension is smaller or equal than 4, the synchronization cannot be thermodynamically stable. So let me now mention how topology affects dynamics. So you know, topology is the study of shapes and shape invariant. What happens when you deform a shape, right? And what remains the same. And what remains the same are invariants such as the Betti number. So for instance, in a network, the number of connected components is beta 0. And the number of independent cycle is beta 1. And if you have a sphere, you have a beta 2, a cavity, a two-dimensional cavity inside. So in a network like with this fungal network, it's clear that the presence of cycle are an important property of this network, and there are tools, including persistent homology, to characterize the structure, the topology of the network. But we don't want to cover this here. What I want to cover is how topology can describe dynamics, and in particular, topological signals, so signals not only defined on the nodes, but also on the links of the network, and if you have a simplicial complex, also on triangles and so on. So these topological signals are, for instance, synaptic signal between two neurons, or for instance, current at different location in the ocean, or flux symbological transportation network. And if you want to have an example of topological signals on triangles, for instance, you can take the citation of a team of authors. So you have a team of 3 authors and you have number of citations associated to the team, so the triangle. So these topological signals are treated with matrices, which are representing boundary operator, and this boundary operator map every simplex to the simplices at this boundary. So for instance, if you have a triangle, it maps the triangle to the 3 link at the boundary, where you go around the triangle with the same orientation. So these boundary operator are matrices formed by node and links and triangles and so on. And for instance, for each triangle, it will give you 1, 2, 2, 3, and -3, 1, right? Because you want to go around this. So this is the boundary. And so they can be interpreted as a discrete divergence, discrete gradient, and discrete curl of signals. So from this boundary operator, you can describe Hodge Laplacian. This Hodge Laplacian describes diffusion from n simplices to n simplices through n-1 or n+1 simplices. So if you are on links, you can go from links to links through nodes, or from links to links through triangle. And interesting, the dimension of the kernel of this Laplacian are the Betti numbers. So these Laplacian are algebraic objects which link to the topology of your object. And they have this property, this Laplacian, that if you have a signals on the links, you can decompose in a unique way into a gradient flow glowing toward the sinks or away from a source. In a network, they can go around the cavity and they can go have curl flow. So this they describe signals of a given dimension, but you can also use the Dirac operator, which links signals of different dimension of node and links and triangles and so on. So the question is, how can we use this to describe dynamics? And for instance, if I want a dynamics that describe the collective phenomena of topological signals, so can we have a Kuramoto model of signals defined on links? And then you realize that in terms of this boundary operator, the standard Kuramoto model, which I described before, can be written just in this way. And it is immediate to see that the higher order Kuramoto model will make use of boundary operator of dimension n and n+1. So you couple each link to the link that share nodes, but also to the links that share triangles. And this dynamics is such that the synchronization, the collective phenomena, is the collective phenomena that goes around the cavity. So it's a collective phenomena that is affected by topology in the sense that is affected by the cavity structure of the network. So it's a new way in which topology can affect dynamics. And the other mode, these modes are free to move, but the synchronization occur if practically the other mode all freeze, and this remains the only mode that are dynamically persistent. And in fact, the other mode, they do freeze, and you can do that by considering the projection on triangle and on node, and you can find the order parameter, and I see that the synchronization occur at zero coupling. So these cavities start to emerge as the dynamics occurring in your simplicial complex on all these cavity of the simplicial complex. But you can also couple these projected dynamics, and what emerged there is that you can have always this synchronization around cavity, but it can emerge at a non-zero and discontinuous phase transition. non-zero coupling and in a way that is discontinuous. And we check this on models, but also on real data, finding good result. So we also couple node and links dynamics, finding this synchronization transition occurring simultaneously on node and links. And we have good theory, which fits well the result on random graph. And we can also use the Dirac operator, which allow us to do this coupling, but in a local way. While before I was mentioning a way to do the coupling, but with a global way, by a knowledge of the global dynamics of the system, it is possible also to induce this continuous transition also with a local coupling using the Dirac operator. And this Dirac operator, this Dirac synchronization actually leads to an emergence of some rhythms that are low frequency, and they are there also in the frame in which the intrinsic frequency are zero. So practically is a rhythm that emerge from the topology of the network. So we can also use this Dirac operator to kind of filter out signals on node and links. This is a more of a machine learning kind of application. Finally, I want to cover this direction, how combinatorial and statistical property also in higher order network affect dynamics. And this is an exciting research direction which involves triadic interaction. This is when you have one node which affects the interaction between other two nodes. So this interaction can be signed, can be positive or negative. So you can have in ecology one species that affects the interaction between other two species in a positive, in an enhancing way, or in an inhibiting way. And these are very popular actually in ecology, but less popular and less studied from the theoretical perspective. So what we want to do is to study percolation in presence of triadic interaction. So percolation is the study of what happens when you damage the links with probability 1 minus p, and then you look here, all the nodes are connected. Here, the largest connected component is reduced, and you monitor the size of the giant of this largest component, which is also called the giant component. So you monitor R, the size of the giant component, as a function of P. And in random graph, for instance, you go from something that has a large giant component to samples in which the giant component disappear at the percolation threshold. This is on finite infinite network, of course. But this is a second order phase transition, very well studied, very important. Now let's see what happens with triadic interaction. So we have structural links, node connected to node with structural links, but some of the nodes also have the ability to regulate the presence of a link between other two nodes. And they can be positive or negative regulation. So we assume that the activity of the node depends on if they are in the giant component or not. So nodes are active if they are in the giant component, and the regulatory interaction are active if they are connected at least to one active positive regulator node, and none of the active negative regulator node. And then the model is like that. Step one, we calculate the giant component. All the nodes in the giant component are active. Then we deactivate all the links that are connected to at least one active negative regulator node, or that they are not connected to any active positive regulator node. And then we also introduce some stochasticity. The other links are still damaged with probability 1 minus p, such as in standard percolation. And then we repeat from stage one. So here you have a toy model. So all the nodes are in the giant component. So this node is a negative regulator of this link. So this link is damaged at the next step. Since these two links are damaged at this step, these nodes are not any longer in the giant component. And you can go on, and since this is a positive regulator, now you damage this link, and you note that at time t equal to 1, you have the same configuration that you have at time 4. So the network is blinking. The giant component is switching on and off. And this is also in the theory, this is the theory and these are the simulation. The giant component in time goes from 80% to 50%. So you have something like this network that turns on and off in time with a period 2. But actually, it's not only like that. If you change the parameter of the model, you can have period doubling or even chaos. So the giant component is now chaotic variable, dynamical variable, chaotic variable. And so you can have your network with changing time in a very chaotic way. So the giant component is, the percolation now becomes a fully fledged dynamical process, which you have this dynamics of the giant component. And the phase diagram, which was this second order phase diagram in which you have the giant component as a function of p, becomes an orbit diagram in which for some value of p you have 2 giant components, for some value of p you have 4, and in some region you have many. And these are the theoretical prediction with just kind of mean field approach, and these are the Monte Carlo simulation on 10,000 nodes. So the theory captures very well, despite the simplicity, captures very well the phenomenon. We also use some real data. The theory only valid for random graph, but we can do the simulation also on real data, and we see that this phenomenon still can persist. So we take a real network and then we place random regulatory interaction. In absence of negative regulation, instead, the dynamics always reach a steady state. So there is no anymore, if there are no negative regulation, there is no anymore this chaotic dynamics. But the transition is still interesting because it's discontinuous and hybrid. So this triadic interaction can actually be extended to hypergraph. So you can have a node regulating an hyperedge, or to multiplex network. So you can have a node in one layer regulating the link of another layer, and we are working on that. So I reached my conclusion. In presence of higher order interaction, the interplay between structure and dynamics is mediated not only by the combinatorial and statistical property, but also by topology and geometry. And this is opening a lot of interesting scenarios, in particular in the study of topological signals, which can undergo collective phenomena, such as this explosive and rhythmic synchronization, also direct synchronization. And then we have also that this triadic interaction inspired both by ecology and by neuron-glia interaction can turn percolation in a fully fledged dynamical process when really the giant component itself becomes a dynamical variable that changes in time. So these are some of the indications of the references in which practically we combine topology with dynamical signal for this higher order network. And yes, so this is the main message of my work, and these are my collaborators. Thank you so much for your attention.
Thank you so much, Ginestra, for this talk. And I think I speak for everyone saying we admire, we're so thankful that we have you here and that you gave your talk, even in spite of of not feeling quite so well. We are extremely grateful for your presentation. Let's see. I think if anyone wants to, let's see. I'm bringing up my list of participants. If anyone wants to put their questions in the chat, or if anyone wants to raise their hand, they're free to do so. But I think
I've raised my hand several times.
Oh, I'm sorry. There you are. I don't have my have it raised here. Please feel free.
Yeah, thank you for the talk, Ginestra. I was wondering about the three-body interactions which you have, so you call them triad. I mean, there are many different types of three-body interactions, and yeah, your results, how much do they depend on the specific form?
Yes, so I think sorry, let me just try to yeah, I would like to share my screen, but going to some previous slides. Yeah, I think I presented this slide, right, in which I classified this different type of interaction into hypergraph, simplicial complex, and network with triadic interaction. But really, the story is that you can easily
but let's say you are a network with triadic interactions. Even there are many different types of interactions. I mean, you choose a nice specific form, but you could have completely different type of triadic interactions. So
you mean the regulation, the dynamics?
No, I mean, yeah, I mean, it's a three-body interaction could be very different. I mean, you have a specific example. This is positive and negative regulations. You could have many other possibilities. So,
yes. So I think first of all, the logic that we use can be generalized. I mean, we choose this simple logic that you need at least one positive and none of the negative, and this, of course, is straightforward. It's clear that this can be a Boolean kind of logic. And in terms of also what kind of effect one node that regulates an interaction can have on the interaction itself, of course, it might not just be percolation. It might not just be removing a link. It might be also something else. For instance, I don't know, coupling, changing the Jij of an easy model, right? So, but in terms of structure, it still remains that is a kind of a node affecting a link or an hyperedge. And I think this is interesting, although you might always claim that this is always reducible to a factor graph or a hypergraph, in the sense that you take as node either the nodes or the structural links. And then practically, if both the node and the structural links are nodes, then you have kind of a network with two types of interaction, right? The solid line and the dashed line. So, and also a simplicial complex is a particular kind of hypergraph you can claim, because the simplicial complex is formed by simplices, so that are indicating many bad interaction between 4, 3, many nodes, but they have the property that they should include this set of higher order interaction. If you are such that if you include a triangle, then you need to include all the links and all the nodes of the triangle, because when you do this boundary, you want that this is the thing that you want to do, the boundary of the triangle. You need to have the links at the boundary, so the system must be closed under the inclusion of these links. But you can claim this is an additional property that you are imposing to an hypergraph, so that actually hypergraphs are the most general structure. And then, of course, this different variation allows you to treat more topology, more topological property, or to, just regulate some of the interaction, which I think it's quite inspiring, this triadic interaction, because it can practically determine, can be key to determine higher order causality. So when a node causes something in an interaction, to an interaction, so with triangle is typically not easy to find a meaning of causality. So if you have an XOR function, you have that any node, any two pair of nodes can determine the third, but where the flow of information goes from where to where, from which pair, it's not clear. So
Thank you.
Don Burke, you've had your hand up for a while. Thank you for being patient.
Yes, thank you very much. I'm going to ask this question with great trepidation. I'm an experimentalist at heart, and so let's see. At the risk that you might actually answer the question, I'll try asking it. When we think about genetic circuits in engineered biology or in natural biology, we often think about some of the simpler behaviors that you described, such as the stable oscillation. I was really fascinated by the chaotic oscillations that you were getting toward the end. And it was somehow reminiscent of, for example, if you have either lots of genes available that are not always turned on, but something's turning them on or off, or lots of resources in the environment, chemical compounds, for example, that some organisms could use, but those organisms aren't present at the moment. So I can imagine how to go from what you described to the types of biological systems that I'm used to thinking of. But I get lost in terms of trying to understand what the rules are. Could you please remind us what it is about the topology of the system that supports this sort of chaotic behavior?
Yeah. So you damage the percolation, you damage the links, and then you monitor the giant component. No? Then when you do this step, you take all these nodes in the giant component and you set it active, while these other are inactive. This is the first step. Then the second step is that there are regulatory interaction. So if this is active and this is a negative regulator, this link is damaged at the next step. But if this is a negative regulator and it is becoming inactive, in some steps, it allows this node to be still there, and these links to be still there. And these links can be still there with probability P. Okay. And similarly, this is a positive regulator. If this is on, this link can be on with probability P. And if this link is off, this is a positive regulator, so this link will be deterministically off. So this is the example. All the nodes are initially on. So this is a negative regulator of this link. So this link is off. And this link is off at the next step. And this is a positive regulator, so this link is on. Okay. So since you are cutting these two links, these two nodes are not in the giant component anymore. So at the next step, they will not be active. Okay. So now this node is not active, but it's a positive regulator. So this link will be damaged at the next step. And
right. I mean, you're using the term damaged. I would probably, in my field, I'd probably use the term the gene is on or the gene is off. The transcription is happening. The protein is phosphorylated. These are, or there's a drought and the pond is frozen. I mean, there's so many ways of doing that. This is fine.
Yes, you are correct. This is not damaging. This is regulating.
Yeah.
Interaction, because it's not damaging forever. It's regulating down, then it can turn on.
I mean, the context of an origins of life, I could easily imagine a potential network exhibiting just a stable behavior, an oscillating behavior, stably oscillating, chaotically oscillating, or simply dissipating, which would not really be an origin of life. It'd be a frustrated origin of life. And I'm trying to understand what the topological differences among those categories might be.
Yeah, so dissipating means that you don't have a giant component.
Yeah, exactly.
There is this kind of possibility, so that you don't have giant component, or you start to have it. Here, this is occurring at a discontinuous transition. So either you don't have it, or you would have it at a given, 15% of the node. And then you might have two solutions, so a period 2, and then you can have a period 4, or you can have this chaotic behavior, but then if P is becoming larger, you have some ordering again. So this is
nice. Thank you.
Wonderful. Yeah, I had a response to your question, Donald. I put it in the chat for everyone. I think this really gets to the promise of, and network modelers and theorists have known this for quite a long time, that the rules of the system may be different from discipline to discipline, or the subject of study may be different, but learning how to master and understand and describe the underlying dynamics in these very large multi-component systems is, a potentially universal approach that can be leveraged as much for gene regulation as for complex chemical reaction networks, or if you have a particular setup with a hydrologic drainage or something like that, or with these really small vesicles that might be interconnected and might have some coupling between chemical synthesis and, say, mass flow. All of this becomes right to the surface, and it's just a matter of how you phrase it and how you encode the rules into the system. All right, I didn't see any other questions, but once again, I want to thank Professor Bianconi for joining us, and I want to emphasize to everyone that this work is Professor Bianconi is recognized as a world leader in these kind of topological explorations of what a system is and how it can be described, and the mathematics that can arise because of it. And so if you have questions or ways in which you think you might be able to apply this work, please reach out to her or reach out to me afterward, and we can find some way to put you in touch. So with that, we'll leave you to your day, and we hope you feel much better. Thank you so much.
Thank you so much. Thank you.
All right. Our next speaker is Dmitry Krotov. He'll be joining us. There he is. And he'll be talking, this is a wonderful introduction to Dmitry's talk, because he's going to be talking about, I believe, Hopfield networks and ways in which networks might have some adaptability or plasticity or learning capacity. And with that, I'll turn it over to Dmitry.
Hi, can you guys hear me? Can you see my slides?
Yes, we can.
Okay, great. Yes. Well, Zach, first of all, thanks a lot for the invitation. I want to start this presentation by briefly summarizing the kinds of topics that I'm working on. So in general, I'm a theoretical physicist by training, and the kind of questions that I'm interested in, they have the following flavor. I like to look at natural physical systems or biological systems and try to sort of abstract their computational strategies that underlie their behavior. And sometimes if we are successful in doing so, and we can actually extract those computational principles from the behavior of natural systems, we can try to construct artificial systems that somehow behaves according to similar principles. And often these new artificial systems, they behave in some sense more favorably compared to their more traditional counterparts that are derived from the perspective of theoretical computer science. So at a slightly more detailed level, what Zach asked me to tell you about is Hopfield networks. And I'm sure many of you have heard about Hopfield networks, right? They have been very popular in the 80s and 90s among people who study complex systems, AI, machine learning, computer science, theoretical neuroscience, etc. But it turns out that there has always been a bit of a problem associated with those old Hopfield networks from the 80s. And roughly speaking, the problem is the following. It turns out that there is only a very small amount of information that you can write down in those old Hopfield networks. And what I will tell you today is some recent progress that has happened in this field in the past 5 or so years, when people realize that you can easily modify the old Hopfield networks in a way so that you can dramatically increase their memory storage capacity. And interestingly, the solution has very much is somewhat similar to what was discussed in the previous talk. Essentially, you need to include hypergraphs and higher order networks, higher order interactions between multiple neurons or multiple nodes. So I'm actually very glad that my talk follows the previous talk because they're somewhat related in that sense. So in order to motivate this discussion, especially connecting it with the topic of this workshop, I want to start by stating the following general question. So how can we build a neural network, or in general, some nonlinear dynamical system that is capable of assembling simple primitive building blocks into highly complex and diverse patterns, right? And in order to make this question a little bit more specific, let me give you one example. So imagine that we have a bunch of patterns. Do you see my mouse, by the way, if I move it? Okay, good. Yes, so imagine that we have a bunch of complex patterns, and let's think about complex patterns as images of handwritten digits. You can see them on the right. And let's say that we try to construct a dynamical system that is capable of assembling those complex patterns from simple primitive building blocks. And on the left, you can see examples of such blocks. So you have 25 building blocks, and each little square here is a little square of 7 by 7 pixels. And each of the patterns on the right is a square 28 by 28 pixels. So you have, in order to assemble one of these handwritten digits, you need to tile together 4 patterns of this size in a row and in the column, right? And of course, you can do it in various possible ways, and this diversity in terms of how you combine those simple blocks, it results in the whole variety of complex patterns that you can see on the right. So the question is, can we build a mathematical system that does something like that or not? And I will try to convince you that this new Hopfield network is actually a very natural framework for solving these kinds of problems. So before I go into the new Hopfield networks, let me start by briefly describing what a standard, most sort of conventional, classical Hopfield network of associative memory is. So in the most conventional form, Hopfield network is a neural network, and the unusual aspect of that of this specific neural network is that it has something that is called an energy function. And in the slide, you can see a cartoon of the energy function shown by this blue curve. So essentially, it's some kind of rugged landscape, and that rugged landscape has a bunch of local minima that are called memories. And these memories are denoted here by these vectors xi1, xi2, xi3, and xi4. So each of these xi's is a high-dimensional vector, and the index, the vector index of that vector is suppressed here. So I'm just simply not writing it. So the idea of Hopfield networks is that you can start from some initial state that is high up in the energy landscape. And then you can let that ball roll down the hill along the energy landscape until it reaches one of the local minima. And when that happens, we can say that associative memory, this network, it recalled the pattern that is located at the bottom of this local minimum. And that's why it is called memory, because the initial state of the network sort of reminds you about some pattern. And then when the ball stops rolling and reaches the local minimum, you can say that the pattern is revealed. And the reason why it's called an associative memory is because essentially the initial hint that you give to the network, where the ball was initially located, sort of is associated with the final state of the network, where the fixed point of the dynamics is, right? So let me give you a couple of examples of how these kinds of ideas can be used in practice. For example, I can give to the neural network as an initial state an image that looks like this. It's a pretty complex, high-resolution image, and you can see that there is a beak of a bird in the left corner, but 3/4 of the image are occluded. So if I let this ball roll down the hill along the energy landscape, you can see that associative memory can reveal a plausible autocompletion of the remaining parts of the image. And that's what the memory retrieval is. So it's a dynamical process, and as the dynamics progresses, we see the stored pattern more and more clearly. So very similar idea is related to denoising. Again, I can start with some highly noisy image on the right. So it's so noisy that you can't even see what is plotted in it. But if I give that noisy image to the associative memory, you can see that the network is capable of removing the noise and reconstructing a nice image of the boat. And if I take this simulation and I stop it somewhere in the middle, hopefully you can see the shadow of the same bird in the background, right? And that's kind of, a general intuitive illustration about how these networks work. So in order to recover a given memory or a given pattern, you need to look at all possible memories, all possible patterns. And the network dynamically decides which pattern is the best match for the initial hit. So it's a highly collective process, and it's a highly nonlinear process of recovering those patterns. Okay, so now let me try to summarize these ideas in a slightly more mathematically defined way. So on the left, you can see the canonical description of the classical Hopfield network from the 80s, given by John Hopfield in his famous paper from 1982. And his idea was that you can think about this system as a kind of like Ising-like system with binary variable sigma I. So here I have a network of capital N computational elements. In neuroscience, they're called neurons, but it doesn't really matter. They have nothing to do with biological neurons in the first approximation. You can think about them as some kind of simple computational units that collect inputs from other units and then decide on their own state. So it's a very, very high level inspiration from neuroscience. It's more like we're doing math here, right? And the energy function for this system is described by this formula. So it's essentially a quadratic form defined with some symmetric matrix Tij. And if we have the set of memories denoted here by these vectors psi mu, so index mu here is exactly the same index as this upper index mu that I used on the previous slide. So psi1 corresponds to the bird, psi2 corresponds to the boat, psi3 corresponds to the cat, etc., etc. But now there is also an extra vector index I that denotes the projection of these patterns into the space that these patterns are defined. So for example, if you're working with pixels, images and pixels, I would be the number of the pixel, right? And psi would be the value of that pixel. So that's the Hopfield energy. And it's a very nice model of associative memory in the following sense, that you can take this network, you can pick one computational unit at a time, and you can compute the energy of the entire network with all the units clumped to their current state, and this specific unit being in the on state or in the off state. And you can pick the smallest of the two energies and flip the corresponding spin accordingly. So if you do that, eventually you're guaranteed to reach a local minimum of the energy function. But sometimes this specific local minimum will not correspond to the memory. And specifically, what Hopfield has shown is that, and many others in the 80s and 90s, is that for this simple network, the best that you can do is the following. You can store at maximal 0.14 n number of memories. So n is the number of computational elements in the network, and 0.14 is some numerical constant that comes out of some statistical physics computation. So what is important is that the number of memories that this network can store scales linearly with the size of the network. And that's, if you think about this, is a huge limitation for what you can possibly do with these kinds of networks. For example, let me give you the following example. Imagine that you are working with binary black and white images of the size 30 by 30 pixels, right? So you have like 900 visible neurons. So what this bound would tell you that there is no way that you would be able to store more than, roughly speaking, 100 of different patterns. And clearly that's just not enough. If you think, for example, about an educated Japanese person who can read kanji characters, they need to know, like, roughly speaking, 5,000 kanji characters to read a newspaper, right? And at the same time, you can easily, digitize kanji characters in the matrix of 30 by 30 pixels. They would be quite recognizable to humans. And what this, mathematical bound tells you that this simple quadratic Hopfield network simply is unable to store that number of distinct memories. So when we started thinking about this, we asked the following question: can we somehow change something in this general construction so that this annoying bound is removed? And that's how dense associative memories appeared. So essentially, in the dense associative memory, the idea is that you need to modify the energy function to make it more nonlinear. And again, I want to emphasize the relationship to the previous talk, because essentially what I want to do is, instead of considering a quadratic form for the energy function, I'm going to include higher order interactions between many Ising spin sigma i's, or as neuroscientists call them, many neurons. And if you do it in a very smart way, so essentially what you need to do is this: you need to take the dot product between the state vector sigma and each of the memories, and then you need to pass that overlap through some highly nonlinear activation function. So that activation function could be a power function, for example, or even an exponential function. So notice that if you're working with the power functions, and if the power is equal to 2, if little n is equal to 2, then this dense associative memory is identically reduced to the classical Hopfield network from the 80s. But if n is bigger than 2, then it's a completely different network. For example, for n equals 3, the energy function would be cubic in sigmas. For n equals 5, it would be it would have a power 5 interaction, etc., etc. So now it turns out that if you consider these higher order Hopfield networks, these days they're called modern Hopfield networks or dense associative memories, you can show that you can store either a power. So you can either have a power law for the memory capacity of this network, or you can even have an exponential storage capacity in the number of neurons. And that's, of course, much more favorable scaling compared to the linear scaling that we are familiar with from the study of the classical Hopfield networks. So if you want to read about these specific developments, you can take a look at one of these two papers, either my paper with Hopfield from 2016 or another very nice paper by Dmitry and co-authors from 2017. So let me give you a simple, intuitive idea of why this should work. And the idea is actually very simple. So the reason why classical Hopfield networks, they get confused when you overload them with a large number of memories is the following. Sometimes you can have two local minima that are located very closely to each other. And if they're too close to each other, instead of having two separate local minima for each memory, you end up having one local minimum in between. And that one local minimum, it's just not informative of either one of the two memories that you are trying to store. And that only happens if the network is not sufficiently nonlinear. But if you make it nonlinear enough, if you allow this flexibility for the shape of the energy landscape, you can actually dedicate a separate distinct basin of attraction for each memory. And that's the idea behind dense associative memory. So essentially, like the difference between the energy landscapes is clear from this simple cartoon. So far, I've been talking about binary Hopfield networks, right? So the variable sigma I is either +1 or -1. But in order to do meaningful AI with these kind of networks, we need to make them continuous. And I don't really have a lot of time during this presentation to define how we do continuous Hopfield networks. But the idea, I want to sort of summarize the general idea, and you can read more about this, for example, in this paper. So the idea is that you need to write a system of nonlinear differential equations. So now, instead of having binary variables, we have continuous variables, v and h, and they all depend on time. And in principle, this is a highly nonlinear system of coupled differential equations that in principle can have a lot of sophisticated behaviors, like fixed points, various complicated attractors, chaotic behavior, etc., etc. But it turns out that there is a very easy way to eliminate all those complex behaviors and keep only the fixed points. And the reason why we want to keep the fixed points only is because we want to have an associative memory. We want the system to start somewhere and then roll down the hill along the energy landscape a little bit and then stop at the fixed point. And the idea here is that you can introduce what is called the Lagrangian function for this system. So essentially, it turns out that every set of neurons in this network can be described using the Lagrangian function for this network. And then if you know the Lagrangian function, you can unambiguously write down the system of coupled differential equations, and you can derive a global Lyapunov function that is bounded from below and that is monotonically decreasing on the temporal dynamics. Again, I don't really want to spend too much time discussing this, but I just wanted to summarize this idea at a high level. And if you want to learn more about this, please check out this paper from 2021. So coming back to the question that I raised at the beginning of my talk, how can we indeed start with something simple and assemble something more complex? So what we can do is this: we can assemble this. We can consider this simple hierarchical Hopfield network. So essentially, the idea here is that now you have two Hopfield networks. One lives between these two layers, and the other one lives between these two layers. And but at the same time, the right way to think about this whole system is one gigantic Hopfield network, and kind of, you know, the mathematical description of these systems can be found in this preprint that is cited at the bottom. So it turns out that if you take this system and you train it on, say, handwritten MNIST digits in the following way. So at any given moment of time, you take an image, you present it to the network, but instead of showing the full image, you occlude part of the image. And the occluded part is shown in the first column here. So you can see like that it's not like a smooth digit, it's like a very dotted image, right? So I randomly dropped some of the pixels. And then you give it to the neural network, you ask the network to arrive at the fixed point, given this occluded hint as the initial state. And then the network produces some kind of output. And then you adjust the parameters of this network in order so that the output that the network has produced matches the actual ground truth unoccluded image that you want to work with. And if you do it for a while, then the network learns that you need to have, roughly speaking, you know, like, I mean, 25 is an arbitrary number, it's a hyperparameter, but for 25 building blocks, it actually works quite well. So you need to have like a small number of building blocks, and you can see those what these building blocks look like here. And you also need to have a lot of assembly rules, how you put those building blocks together in order to form a digit. And it turns out that the building blocks are encoded in this set of synaptic weights that are located right here between the visible layer and the first hidden layer. And the assembly rules, they are encoded in the other synaptic matrix that is located right here between the first hidden layer and the second hidden layer. And that assembly rule matrix is extremely sparse matrix. And again, we don't really tell the network what these assembly rules are. They just simply learn those assembly rules by looking at a bunch of examples of the data. That's like how we generally do things in machine learning these days, right? And this is what this network does eventually. So essentially, like you can see like pairs of images, the initial image, the occluded image, and the final reconstructed image when the network has been trained. And you can see that these reconstructed images, they look pretty decent. So they're certainly like recognizable MNIST digits. They're not perfect. You can certainly tell that they're not, photographs of real handwritten digits, but they do a pretty decent job in assembling this, highly diverse set of patterns. And again, the cool thing is that the number of building blocks is very small, and the number of assembly rules is also quite limited. And we can simply learn all of those from the data. So I've been talking so far about this simple MNIST data set, right? And of course, like it's a highly toy model example. So like, I mean, you can't really publish any paper these days in any of the AI conferences by showing results on these simple data sets. But it turns out that with a decent amount of engineering efforts, you can actually scale up these ideas to, pretty impressive AI systems that work with high resolution images, that can work with text, they can work with graphs, etc., etc. And the idea is very similar. And I'm going to show you how, like, one of such systems that we have built recently works on images. So again, at the initial moment of time, we provide a seed to the neural network that looks something like this. So we take an image, this is the ground truth image, and we occlude some parts of those images. And then in the middle, as I will start playing this movie, you will see how the image evolves in time. And what is cool here is that this specific image that you will see, it has never been shown to the network before. So it has never seen this specific wolf or dog or whatever that animal is during training time. So it's any image you could have sent me like some some of your images, and I could have run my network on those new images as well. But as you will see, and let me just play it, the network does a very decent job in auto-completing this completely new image that it had never seen before. And that indicates that indeed the network has learned a general set of rules about what a plausible set of images might look like. So for example, it knows that if you see a bit of a leg here and you see a body over there, then there gotta be a continuation of that leg, right? And the continuation should, roughly speaking, match the color of the entire animal. So if you show a face and you occlude half of the face, the network knows that there got to be another half of the face, and that another half of the face should be located in a proper location and with proper proportions, etc., etc. So there is a lot of this kind of knowledge that the network is able to learn by simply look at a huge amount of training data that represents a meaningful distribution of natural images. But again, like how we are testing those networks, we are testing them on completely different kinds of images. And yet they work quite well. So I guess what I want you to take from this slide is that the ideas that I sort of presented at the previous slides, they're not only about simple toy model datasets like handwritten digits, but they can be scaled up to something quite interesting and perhaps relevant to the complex systems community, and perhaps is relevant even to the origin of life. Because in the origin of life, in some sense, what we want to do is we want to compose, we want to construct complex chemical compounds, right, out of simple building blocks. And the cool thing about this particular framework that it also has a little bit of sort of biophysics or biochemistry in the background, because it's an energy optimization. So these compounds, these chemical compounds, they in fact correspond to the memorized patterns of the dynamical system that is governed by the overall Lyapunov function. So in that sense, there might be some interesting relationships between the kinds of questions that you guys are interested in and the kinds of questions that we work on in the field of AI. Okay, so let me let me only briefly mention this, because again, I don't really want to spend too much time talking about this. So you've probably heard about transformers. Transformers are like super hot. Everyone is working on transformers. Pretty much all like the buzzwords like GPT-3, like all these language models, large language models that we read about in New York Times, etc., etc. They have the underlying architecture that is a transformer. And it turns out that the kinds of networks that I have described to you, these new dense associative memory, new Hopfield networks, they are in some sense a more general framework for transformers. So it turns out that you can take dense associative memories and you can pick certain activation functions in certain way, and you can mathematically show that they are equivalent to transformers. And that's kind of, interesting because on one hand, it provides you a theoretical perspective on transformers that is by and large missing. Usually, like practitioners, they experiment with transformers by trial and errors. And on the other hand, in the field of Hopfield networks, we do have a fair amount of mathematical theory. So we know, we can compute something, we can argue about their memory storage capacity, about how that memory storage capacity is related to computation, etc., etc. And that's valuable for interpreting what transformers are doing as well. So again, I don't want to spend too much time talking about this, and somehow, sorry, I put my conclusions far away. Where did I put my conclusions? Yes, these are my conclusions. So let me jump to the conclusions. So essentially, what I'm saying is that the annoying memory storage limitations of the classical Hopfield networks that people used to deal with in the 80s and 90s today are gone. So you can store as much information in the Hopfield networks as you want. So modern Hopfield networks can be both continuous and binary. There's nothing that would restrict you to the binary case only. The easiest way to think about these networks mathematically is through the prism of the Lagrangian functions. So it turns out that you can literally pick the Lagrangian for every neuron or for every layer of neurons, and that would completely define the global Lyapunov function, the nonlinear dynamics, etc., etc. And you can also construct deep hierarchical Hopfield networks with many layers of hierarchical representations and various inductive biases like convolutions, etc., etc. And I really think that there could be interesting angle for exploring these new ideas from the perspective of complex systems, because indeed they sort of do have the right flavor for constructing complexity out of simple building blocks. So if any of you guys have any ideas about that, I would love to chat about this. Yes, so thank you.
Thank you, Dmitry. That was wonderful. It's very helpful to get this kind of overview of how these kinds of networks can be applied to, say when we have missing data or we're trying to infer what's going on. It's extremely helpful because as a specific case, as we go toward systems chemistry approaches, it's pretty common that we don't have complete data on what actually occurred in the system. And we're really looking right now for any tool that might help provide some insights into reconstructing what actually happened. There's Donald put a question in the chat. And
maybe you can just unmute yourself and ask. I think it's easier if we discuss.
At least interaction is always better.
I'm here, but I'm running around trying to go teach my class in a few minutes. So I just wanted to kind of put it in there.
Okay.
So
oh, did you want Donald? Did you want me to read your question then in lieu of being here?
Well, let's start there, and I'll just try to not to make too much noise in the background.
Okay, let me just read it. Associative memory seems like a powerful strategy to cut through initial fog and start to move toward local minimum. But it also seems like it might be exquisitely sensitive to initial choices in the moving towards potential solution. Visual puzzles are fun ways to misinterpret partial images. Is there some way to protect the strategy from falling into traps of confirmation bias? Well, I mean, yeah, certainly there is such a problem, right? So if you, and frankly, it's not even only the problem with Hopfield networks, it's a problem with any kind of complex network these days, trained with backpropagation. So as I'm sure you know very well, right, there are these adversarial examples. So you can like take an image and you know, the image, it's an image of a cat and it's recognized as an image of a cat. And then you can take that image and you can change the intensity of a couple of pixels. And then you still see it as a cat. You can't even tell a difference between the altered image and the initial image, but the neural network thinks it's an airplane now, right? And that's a common problem. So Hopfield networks are not immune to that. However, interestingly, they're much more robust to these kinds of alterations. And in fact, we written a paper about this, I think, around 2018, where we literally show that indeed, if you take the standard classification pipeline and you consider, for example, like a standard feed-forward neural network, and on one hand, and on the other hand, you consider these kinds of new Hopfield network, which one would be more adversarially robust? And it turns out that the Hopfield network is actually more adversarially robust. And the reason why is very simple, actually, because it actually helps to clearly delineate the basins of attraction around the proper memories. Because the reason why conventional feed-forward networks are very susceptible to these small perturbations is because the decision boundaries are very brittle. But it turns out if you take these more nonlinear networks and you combine these nonlinearities with the backpropagation while you train them, then the learned representations, they are in some sense much less brittle. And that helps with robustness. So I don't know, Donald, if I addressed your question or not.
Yes, you did. As you were describing the system sort of rolling downhill on this energy landscape, it was very reminiscent of the three-dimensional folding of proteins or of RNA molecules as they go from random chains to structured entities. And it's very common in the physical realm for them to get stuck in local minima because they make some wrong decisions early on.
Yes.
Right. Wrong. It's wrong in the sense of non-productive in getting to the final end. And one of the ways that biology deals with that is to lower the energy barrier between the paths, so that if it does get into those local minima, that it could shift over onto other paths and shake loose and then fall farther down the energy landscape. Is there such a thing as modulating the energy barrier between alternative paths in this associative memory approach?
Yeah, definitely, of course. And that's actually like the role of biology. When you say biology, like make sure that the energy barrier gets smaller, the role of biology is played by the learning mechanism here. So like all these systems, they have a lot of free parameters, like these matrices Xi that I have shown you on the initial slides. We learn them through backpropagation, for example, right? And depending on the values of those weights, those parameters, the barriers can be big, the barriers can be small. But what is cool is that when you make this whole energy landscape, this parameterization of the energy function more nonlinear, it actually helps to better delineate the distinct basins of attraction. And that's a kind of mathematical fact. For that reason, like if you're working in the class of these new Hopfield networks, more nonlinear systems, it actually turns out that the basins of attraction, they sort of makes more sense compared to the basins of attraction in the less nonlinear systems.
If I could milk the metaphor for just one last detail, sometimes when a protein is misfolded, it will trigger the cell to respond to it. Other times when it's misfolded, it is just thrown away. So I'm wondering whether stumbling into the wrong attractor could somehow be recognized by the system. If, for example, you have a chemical network that you're working with and you're saying, oh, I think this is the this is the chemical system that might have given rise to glycoaldehyde, make up something, right? RNA. Then, but there's something wrong about what you've done. Is there something that you can recognize in there and say, uh-oh, I'm on the wrong place? Is there some way I can now shake it loose, but do it in an unsupervised manner so the computer can recognize that?
Right. That's an interesting question. So that would not be part of the framework that I'm describing, but it certainly can be some external feature that is combined with what I'm describing, right? So you can imagine that if indeed you somehow recognize that you are in a wrong local minimum, you can, for example, go back to the network and you can delete that memory, right? Like I can give you an example. Let's say that we train a language model and we scrape a bunch of text from the internet. And we have actually done something quite similar. If you are interested, there is a website, flyvec.org, that you can go out and play with. But then if you download a bunch of text from the internet, often the network learns something that you don't necessarily want to present to people. It just learns a bunch of like offensive language. And then, like if you go to that website that I mentioned, you can actually like click along. Like there is a grid of neurons and you can click on every neuron and you can see what neurons have learned. And then you can see that for some neurons, we don't show what they have learned because they learned offensive language, because people use offensive language on the internet, right? But the cool thing about these kinds of networks is that you can easily identify those memories, those bad memories that you don't want to be part of your final model, and you can simply get rid of them. And that's a very nice property. So, I mean, in the language task that we've played with, we did it by hands, but in principle, you can probably kind of come up with some idea like you don't set in the chemistry world where there is some kind of, external network that detects that there is a problem, and then it can go back to the Hopfield network and say, look, this memory needs to be deleted. Mm. Yeah, so something of that sort. It might be interesting to explore.
Dmitry, we have a couple of questions regarding dynamics in the chat. One is, I think they're related questions in some ways. Are the dynamics dependent on the initial seed, and is the neural network dynamic? Like, the fluxes change from continuous to binary to discontinuous, or is it somehow baked into the setup?
Right. So let me start with the second one. So they can be both binary and continuous, but they're different networks, right? So you need to kind of somehow make a choice. Do you want to work with binary variables, or do you want to work with continuous variables? So the framework allows you to work with both kinds of variables. But once you make the choice, you can no longer switch between the two, right? In the first question, please repeat it one more time. Is the dynamics dependent on the initial seed? Yes, dynamics is of course dependent on the initial seed, right? Because like in these examples that I have given to you, like you can see, for example, that these dynamics in this slide, right? It certainly depends on what you present at the beginning, because technically I could have shown it the image of me here, and it would provide a plausible auto-completion, but because the initial seed was related to this animal, like the whole dynamics is somewhat like localized around that specific animal. So in that sense, it is dependent on the initial seed indeed.
I see. And we've got another kind of general question about whether the Lagrangian that you described satisfies an action principle, and if so, is there a physical interpretation that is attached to it or might be inferred from it?
Right. And sorry, I did not explain this at all. I just only mentioned this. So the answer is no. So there's no, like the systems are not Lagrangian in the sense of Lagrangian mechanics. So there are no Euler-Lagrange equations, nothing of that sort. But the reason why we picked that word for this auxiliary function is because it is related through the Legendre transform to the Lyapunov function. So like in the same way how in the classical mechanics, the energy of the system is related through the Legendre transform with the Lagrangian, in exactly identical way, like the, let me maybe go back to this slide. In exactly identical way, the energy function for this system that you can actually see here is related through the Legendre transform to the Lagrangian function for the layer. So L is the Lagrangian, F is the derivative of the Lagrangian with respect to the dynamical variable, and H is the dynamical variable itself. So it's literally like the Legendre transform. And that's like holds for all kinds of Hopfield networks it's a pretty general statement. So that's why I think Lagrangian function is a very appropriate name for this function L. But there is no action principle.
I see. And I'm going to back out for just a moment, because in one or a couple of your papers, you explored the possibility or covered the topic of whether Hopfield networks are natural or are naturally occurring. And I was wondering if I might pick your brain about, what did you mean in that context? Did you mean in the sense of whether there are actual physical examples of, real chemical systems or real physical systems that might behave like a Hopfield network that are not engineered or designed? Can you unpack those ideas a little bit?
Right. Interesting. That's actually like somewhat of a hot topic in the neuro AI community right now. And so there is this question, right? Hopfield network is a very natural mathematical framework. It's kind of, it's very easily conceptualized, and it's really kind of, at a high level reminds us about how we think about associative memory in psychology. So that's appealing. But then you can ask a deeper question. If we really believe in this kind of math that we have constructed, right? Can you imagine that some sort of similar math describes certain parts of the brain? And that's a much more sort of deep question, right? Because the math dictates certain constraints, how like computational elements, how the neurons are connected to each other, what the weights should look like, etc., etc. And we indeed explored these ideas, and we found some architectural structures that experimentalists know exist in the what is called cortical hippocampal loop. So there's essentially like two areas of the brain, the entorhinal cortex and the hippocampus. And there is like certain anatomical structure between them, the pattern of connection that is sort of reminds us about these kinds of equations. Of course, it doesn't mean that, hippocampus is literally what these equations are doing. And there are like meaningful ways in which these equations deviate from the architecture of that part of the brain. But there is also a good amount of similarities between the two. So that's one idea. Then, interestingly, just a few days ago, we released a preprint on bioRxiv, where we argue that transformers in fact, can be implemented by astrocytes in the brain, right? So it turns out that neurons is only one kind of cells that appear in the brain. There's a ton of glia cells, there's a ton of astrocytes. And in fact, according to some reports, there is even more glia cells in the brain than neurons. And for a long time, people thought that those non-neural cells, they don't really do much. They just, provide essential metabolic functions for neurons to work. But recent experimental data suggests that there is a ton of computation that is happening at the level of astrocytes. And what we did in that preprint is we tried to connect the computational principles of transformers. And transformers are, again, very much related to dense associative memories, the way I introduced them here, and the plausible computations of astrocytes. And again, we see a lot of similarities between what is known about astrocyte computation, the kind of plasticities that they are capable of performing, and the kind of information exchange with neurons and transformers, specifically self-attention operation. So if you're interested, please take a look and let me know what you think, because it's a completely brand new preprint.
We certainly will, and I would encourage everyone here to take a look at it as well. I've got one more quick question before we go to our next speaker. I'm just kind of curious as to whether, it's a related question. In this part of kind of NASA exobiology, astrobiology activities, we're focused mainly on, synthesizing and building and experimenting with systems, but there's a related field that has to do with trying to assess whether a given system, you don't know whether it's abiotic, biotic, prebiotic, or whatever. Whether it's complex or capable, or whether what you read from it is attributable to biology or not. And I was wondering if this approach, like maybe not in terms of, instead of trying to fill in the details when you have occluded data, but you apply, say, a Hopfield network or some other kind of neural network, and you can measure how the system is trying to adapt or trying to connect what is known with what the output is, if that might serve as some kind of approximate metric, something like Kolmogorov complexity for the system, if it's having to activate, say, a large number of rules or generate a large number of neurons to efficiently approximate the system, if that might be like some kind of standard or metric for the complexity of the system.
Right. That's a good question, and I'm going to try to answer. I'm going to try to give you two answers. So first of all, you can ask the kind of questions. I train my network on one set of data, and then I show a new piece of data. Does this new piece of data come from the same distribution, or is it like completely different? Is it from like a different universe? And a natural way to sort of approach that question would be to calculate the energy. And if the energy is low, like literally like this kind of energy, if the energy is low on the new example, right, then it's likely that it comes from the same distribution as you have seen before. If it's high, then it's like a completely different thing. And the second question regarding complexity. So the cool thing again about these networks is that they're much more interpretable compared to pretty much any other network that you can play with in the standard machine learning practitioner's toolbox. And the reason is very simple, because it's somehow, because this nonlinearity that I spent so much time talking about is essentially enforces some form of sparsity in the latent representation. So essentially there is only like a few patterns, the few hidden neurons that are responsible for working with a given data point. And the reason why this is nice is because it provides a lot of potential for interpreting the kind of computation that these networks are doing. So you can literally go inside this network and say, aha, I see that for this specific point, I activate only these 5 neurons, and only 5 and not like 1,000, as you would typically see in any other deep learning network. And again, kind of this indeed can open potential ways of exploring complexity, the patterns of activations, and interpretability aspects of these neural networks in general.
Got it. Thank you so much, Dmitry. Thank you for the extra couple of minutes of your time with answering our questions. If you can stick around to answer a couple of other questions, that would be great. But otherwise, thank you very much for your time.
Yeah, thank you very much for the invitation, Zach. Mm-hmm.
Yes. Next up, we have Madhurima Nath, who is joining us from Slalom Consulting here in the USA. You'll notice that a number of people who were referenced either in talks yesterday and who are also speakers today are finding a lot of applications and working within academia and beyond. And Dr. Nath is one of those examples. So, and with that, I'll turn it over to you.
Hello, Zach. Nice
Hello.
to meet you. Yeah, thank you for the introduction. Let me just share my screen to get us started. Just checking, can everybody see my screen? Perfect. Thank you.
It looks great. Thank you.
Okay, thank you again for inviting me to this platform, and really excited, and hopefully we can all learn a lot from each other. So my talk today is about the work I did during my PhD on kind of developing this algorithm tool called network reliability that I used on a number of diffusive processes on complex networks or interacting systems. And today I'm going to just give you an overview of how I used it and what are the applications I studied and how why is it novel, and I'm going to talk a little bit more about that. So let's see. Okay. So let me start by something which we all have learned in our school, that is ideal gas. And ideal gas can be, we can look at it from, treating the molecules as billiard balls, bouncing off the walls, and that would be looking at it from a microstate level. We can also look at how these bouncing off each other is affecting the macrostate or the state variables, which are pressure, volume, temperature. And we can write down equation of state, draw some plots. And connecting these microstates to macrostates and building up the synthesis is the going from the kinetic theory to the ideal gas equation, which we all know as PV equals nRT. And this is great. This gives us a lot of insight. However, the reason I'm talking about this example is that in physics and in science in general, we always tend to start with the most simple model and the idealistic model, and then start adding perturbations or higher order terms to try to understand what is happening in the real world scenario. So moving on from ideal gas to real gas, we would be adding some interactive terms and try to see, are we reaching what we are seeing in real life? Now, the good thing is that we know how to deal with these kind of problems when we have like less number of molecules or less number of processes interacting with each other. But on the other extreme, we also know how to deal with it in the thermodynamical limit, or 10 to the power of 23, or infinity limit. So, but the main challenge is it has to be very ordered system, very regular system, and we have theories for that. Now, as I said, passing from the ideal case to the real case often involves a lot of many body interactions. And we need a lot more, we have to overcome a lot more challenges to get to this real world scenarios, as there might not be a lot of symmetry, there might not be very well-defined distribution. And as I said, it lies between this uncomfortable place of 10 to the power of 23, and that can be anything. And not to mention a whole lot of interactions going on within each other. So I like to think of it using some simple semantics. So we all know that we were passing through this COVID pandemic. So let's just take this example, right? There are biologists and doctors and other scientists who are looking at the pandemic from a very, let's say, microscopic level, or even like, how does this virus affect the cells? How does this virus affect the organs? And how is that bringing out symptoms in an individual? So that kind of relates the organs and the individual. Then there are social scientists, epidemiologists, computational modelers who are looking at the spread of the virus from an individual to another individual, and then further to a community, and then from a community to a city, and then kind of making the scale larger and larger. So we can think of real world problems also in a similar way. There could be microscopic interactions which affect the outcome, or there could be macroscopic interactions. There could be temporal interactions. So it's a very messy and challenging problem that we are trying to solve. And it's not just limited to physics or biology or chemistry. It can be an interaction of all of these things. So I know there have been a lot of talks, and Zach in the morning introduced that complex systems or complex networks are great to study these kind of applications. We all know that there was a Nobel Prize last year to kind of acknowledge how much complex systems can be useful to study climate change or even fluctuations in physical system, ranging from atomic to planetary scale. So I find complex systems very interesting, and I'm going to talk about how I used graph theory and complex networks in my research. So let's go back to the COVID example, right? So I studied it in this manner. Like I took data or population, people interacting with each other. This could be interaction with friends at school or going to grocery. And I want to mimic this interaction, and so what I would do is put this into a network or a graph. And I'm also capturing the state of me being either sick or susceptible to the disease, or I'm recovered from the disease. So that kind of allows me to capture the state as well as study it over time. Like, am I spreading it to others? And how is that spreading over time? How is the disease spreading over time? And what is my state doing to my neighbors? So I kind of used complex networks and these dynamical nature of this diffusion process in my work to kind of look at how these small interactions between each other affect the outcome. So another example that I looked at, and this is why I like graphs, is these are very abstract. So I can put it in another context, which is, let's say, I'm studying trade of commodities between countries. And so I'm going to take an example. Let's say the commodity I'm trading is potatoes, and potatoes is being going around the entire world from US to Europe to Asia or whatever. And let's say there is a pest which is native to the crops in Spain, but it's an invasive species when it goes to some other country where that pest is not very native. So since it is getting transported along with the trade, now that pest is affecting another country. So how can countries take measures to, not affect their food security, like to prevent and take some steps? So I can kind of model this scenario using graph theory. And in this case, the propagation of the pest or the invasive species would become by dynamics. And I can kind of study it in the same way as I did with people, whether a country is susceptible or whether a country has been infected, and what kind of steps that country as a whole should take to mitigate this spread. Now, all of these systems I talked about they don't have a very well-built ordered structure. So this is where graphs are extremely useful. But I want to kind of bring the attention to what happens when I do have an ordered structure, like I have a lattice, and I can still use graphs to study this thing. And Ising model is a very well-known problem in statistical physics. And what we try to study is, how does the effect of a spin flip at a particular site affect the magnetic property of that system? And what happens if there is an external field? How does this spin flip change? How does the magnetic property change? So again, I like to think of this as the propagation of the spin flips through the lattice. So I can again model these as a diffusion process on networks. And why I am talking about all these examples is that in all these examples, I'm trying to find out the probability that a finite system, which starts with some kind of initial configuration or a specific configuration at certain time t, will have some kind of property later. And this is a very, very generic statement that I just made. As you can see, I can apply it to a lattice of magnetic spins, and what is the probability that two magnets are separated by some k lattice sites. Or I can just try to study, given an infected individual in a susceptible population, what is the probability that there is an epidemic outbreak? I could even extend it, saying like, what is the probability that there exists some kind of path between 2 points, which could be a source and a terminal. And this, I'm giving these examples to kind of convince you that the question I just posed is very generic, and depending on if we have an interacting system and there is some kind of diffusion-like process, I can try to calculate this probability using complex networks, and that's and I'm trying to build a generic tool out of this. So this is basically what I'm going to talk about throughout the next 30 minutes or so, is network reliability. Network reliability was first introduced by Moore and Shannon in the 50s to study electrical circuits. And we kind of took that idea and implemented it on complex networks, or what I like to call it as graph dynamical systems. So the question I posed previously is, I'm trying to find the probability that a finite system at a specific configuration at time right now will have some certain property later. This probability can be calculated using network reliability. And I'll walk you through examples of how this can be calculated. And just a little bit of definition of graph dynamical system is that it's still a graph theory, it's still a complex network, but it's a formal way to capture the structure and the dynamics. And it could be unweighted, it can be weighted, it could be directed edges, directed interaction, and it doesn't have any irregularities or symmetries initially assumed. You can just put it on any kind of problems. And network reliability, as it turns out, is a very generic tool, and it can answer all the questions that I just asked. Like, it can find the probability of a path between two nodes, or estimate the outbreak potential of an epidemic, and so on and so forth. So before I get into how to calculate network reliability, I just want to re-emphasize on why I'm using Moore-Shannon network reliability. It's a very generic tool with a wide number of applications. And one of the things I think it's more useful is that it is a function of both the dynamics and the structure. So here I have just a very schematic of a very small toy model. And you see, without going into any math or definitions, the purple network here has some nodes, and all the lines or edges are connected between these nodes. And the representative reliability curve is the purple curve on the plot. And if I remove even one node out of this network, which is the node between if I remove one edge between the nodes B and D, the network now becomes green, and the corresponding reliability curve is the green curve. So you can see that even removing or even a small change in the structure will have significant change in the reliability. So it captures the dynamics, which is, I'm calling it as X, and I will explain what X is. It's a function of X. Along with that, it's also capturing what changes are happening in the structure. And this is just a mathematical formulation of, if you want to think about what network reliability is, it's just counting how many dynamical paths lead to the configuration that we are interested in. And it has a probability associated with it. So it's just a summation of all this probability and the fraction that has that certain property. Okay, so let's do a little bit of walkthrough on this toy model. So how would I calculate a path that exists between the node S and T using network reliability? And we can put it in the context of, let's say, someone getting infected. It's easier for me to visualize when I have a real world application. So let's say S is infected, and the rest of the nodes are susceptible to this infection. And this is my initial condition. So I want to calculate the probability that the node T at end will also be infected, given that S starts being infected. So here I have a graph dynamical system with 6 nodes, 7 edges. I am going to assume SIR dynamics, which is at time T, if I'm susceptible, at the next time in my simulation, I will be infected, and then at the next time step, I will be recovered. Again, this is again generic. I have worked with SIR, but there are other infectious disease models as well. And X, as I said in the previous slide, X is going to be, in this case, the probability of transmission through each of these edges. So just the boundary, the most extreme conditions, when S is isolated, let's say it's quarantining, there is no way he has any connections with any of the other nodes. So there is no way T is going to be infected. So no path exists. When all the edges are present, then yes, there is going to be a path and T is going to be infected. So these are my kind of extreme cases. What happens when some of the edges are connected, and what are the bare minimum that I need to get from S to T? I could go from S to B to T, and that would require 2 edges. Or I could take the top route and use 3 edges to go from S to T. Now, this kind of gives me, I can write this down in terms of what I call as inclusion-exclusion principle. So that is counting all the edges that contribute to the probability going from S to T, and those which I don't want to overcount it, so I need to subtract those overcounting. So that gives me the expression x squared, 2x cubed, 3x5, and x7. Now, I can also think of it as choosing edges as a combinatorial problem. Like there are 7 edges. If I choose 1, then I'm not connected. If I choose 2, for example, these 2 that I have highlighted here, then yes, I will get a probability of connection between S and T. If I choose 3, 7 choose 3, then these 3 edges that I show here will get me connected and the rest will not. So we can also think of it from a combinatorics perspective. And the nice thing about reliability, which is RG of X here, is that it can be written down and calculated in both these ways. And this kind of helps me calculate reliability as the network gets larger and larger. And I'm going to discuss that as well. So going back to what I just said, I can think of this as counting edges from 1 and 2 and 3 and which of these are connected, and writing it out as like a Taylor expansion, because this is like a Taylor series around x equals to 0. So the plot I have on the right of the slide is basically showing if I consider only certain terms of the entire equation. So how does my Taylor expansion look like? And just keep an eye on the betas, and I am going to explain the betas in just a little bit. But I can also do it in the opposite way. And that is, I want to find out what makes S and T not connected. And I can start taking out edges, as if there will be no edges, there is no connection. If there is one edge, there is no connection. So I can think of it in the opposite way. And that would be expanding the Taylor series from an opposite end, which is at x equal to 1. And the equation becomes much more messier, and the calculation of thinking which edges to remove to, what are the combinations to remove so that S and T are not connected is much, much messier compared to like finding out connection in this particular example. So this is just showing all the curves if I'm taking certain orders of terms in 1 minus x. So I started with saying SNT, I need SNT to be connected. I'm trying to calculate what is the probability of SNT. So why am I now going and saying why SNT is not connected? And the reason is that this is going to help me calculate network reliability from both ends. And this is where I like to use that, take example and from statistical mechanics or statistical physics, where I can use perturbation approach to evaluate network reliability. I can kind of think of it as a weak coupling expansion around x equal to 0 and get some terms, and then a strong coupling expansion around x equal to 1 and get some other terms. And it's very effective in kind of calculating network reliability using this perturbative approach. And this is going to come in handy very pretty soon. So on the left of the screen, you see that I have written down network reliability in terms of betas and a combinatorial factor and x to the power of k and 1 minus x to the power of n minus k, where n is the total number of entities that I have. It could be nodes, it could be edges, but we can't mix it. We have to either keep it in the node level or an edges level. And the reason for me to write it in terms of betas is, it's a very, it kind of goes back into a basis function, which I will discuss in 2 slides. But the reason I want to go into the beta form from these alpha Taylor series expansion is that the moment I impose the duality, then that imposes some unitarity and monotonicity constraints, which I'm not going in detail with, which are not respected by the Taylor expansions. And that's where I need to transform the Taylor series or the power basis into the Bernstein basis. And that's why I kind of started writing it down in terms of the combinatorics and the beta functions. And a little bit about Bernstein functions for before I go any further, is that, as you can see, it's a very natural basis for reliability. The basis function is just n choose k x to the power of k 1 minus x to the power of n minus k. And that is kind of there in network reliability. So it makes sense for us to just use Bernstein basis as the basis function. And there is also a very nice duality property built in the function itself. And the beta k's and beta bar n minus k's are actually equal to each other. So if I know one, I basically know the other end. So I don't have to start, calculating all these cuts and all going through all these complicated formulas. And these are actually used to draw Bezier curves in computer-aided design. So if you have ever, put some points in old Paint or Microsoft Word and try to draw curves between the 2 points, the function that is used behind the screens is actually Bezier functions or Bernstein functions. So they are very useful in drawing, plotting curves or drawing curves if you have the points. So now this slide actually will give you an walkthrough of why I went on to all this trouble of calculating reliability or RG of X from all these 2 different perspectives, like from the not connected side and the connected side. And let's take the same toy model that I had, And let's just consider that I know terms up to order x cube at the end x equal to 0. So that would mean that I only know these 4 alpha k values. And let's say I know some terms, or I have calculated some terms on the other end, which is at 1 minus x end. So that means I know the alpha bar k's for the other Taylor expansion. Now, the good thing is the moment I start changing the basis from Taylor basis to Bernstein basis, you can see I already know a lot of my beta k's, and that kind of helps me estimate what network reliability would be for this toy graph without even knowing the midpoints. And this is exactly what the plot shows here. Is the purple lines are the truncated Taylor series expansions, and the dotted line is the exact formulation. And as I said, Taylor series does not obey all the constraints that is required by network reliability. But changing the basis from Taylor to Bernstein gives me this nice bounded region of Bezier polynomials that fits very well and is a very good approximation, given I know only a couple of terms at the end, for the exact power series. And since it's nice and bounded, I know that my estimation will lie between these bounds, and it provides me with a very nice interpolation. Now, the reason why I'm doing all of this is because the exact evaluation of all network reliability polynomials for any of this realistic system is extremely difficult. It's proven to be a sharply hard problem. For a toy graph, yes, I can sit down and write out all the combinations, but for a larger system, we definitely need Monte Carlo simulations. Now, the argument can be, why can't we do Monte Carlo throughout? Well, like what is the added advantage of using perturbation techniques and all these complicated math and transformation? Monte Carlo simulations are great, and they do really, really well when it's in the, let's say, if I go to the reliability curve, what I call it the middle region. When K, or when the sampling size goes really, really small, then here by K, I mean the number of edges that I'm sampling for the Monte Carlo simulations. When it is really small, then I need to have a large number of samples, and I need to draw a large number of samples to get a very accurate estimate. And I'm trying to kind of make a combination between Monte Carlo simulations and the perturbative approach to make an efficient algorithm for large systems. And that is exactly what is summarized in this slide. Is for an efficient estimation and interpolation, what I do for real world systems, which could be directed or weighted, is I use the perturbative expansion method from statistical mechanics, get some estimates at the ends, then change it to Bernstein basis, and then for the middle portion, use Monte Carlo estimates. And this has worked pretty well for us in our research. So we have been able to apply this to study epidemic outbreaks in Ad Health Friendship Network. And this is a study which was done to kind of with different schools, and they were asked to kind of nominate their friends. And it's a very nice, the literature is out there, how these friendship networks were built. So we took one of those networks and we compared it to a representative network model, which had the same structure, but just mimic. So this kind of shows you not only that reliability can estimate the epidemic potential on networks, but it also tells you that just mimicking only the structure of the network is not enough for a problem like epidemic outbreak. We also need to take into account of the dynamics, and reliability curves actually give you that upfront, saying that, okay, even though they look structurally the same, there could be differences in the dynamics and how this infection spreads. So it not only, it gives you an added information with this curve. So that's why I like to use reliability for these processes. And again, going back to one of the examples that I said is, I also applied reliability, the similar method for kind of figuring out what are those connected clusters of countries where some invasive species or pest could spread. And can I identify which are the vulnerable countries, and can I see, what do I see, basically, it was the question. And the thing is, yes, we can calculate like connected components or identify what the vulnerable countries are. And it will tell you how that happens. And not only that, the result actually concurs with the trade agreement. So you can see those patterns coming up, like USA, Mexico, Canada have a North American Fair Trade Agreement. So they kind of cluster together. So that says that if there is an invasive species or a pest that is, like right now in some crops in Mexico, it is possible that it can come into USA and then it can come into Canada. So it's kind of gives you that additional insight. It also shows you that the plot on the left is actually for the tomato network being traded throughout the world, and the one on the right is for potatoes. And it's very evident from these that tomato is very regionally traded. Like you can see like clusters of continents being, and countries and continents together. Like there is a European cluster, there is an African cluster, there is a North American cluster. But for potatoes, it's very different, since potato is very global and it's also stable. So it's traded globally, and kind of all countries are kind of mixed up, and you will see some Asian countries coming together, some African countries coming together with European countries. So these are additional insights that we can get out of reliability as well, along with what we started with, that is to calculate the probability that we wanted. So I would like to end with, Newton's quotes, is that we do start our analysis to, we can go from, it could be both ways. We can study from effects to their causes, or the other way around. And we want to keep on going till we make it more general, till the model or the synthesis maybe we are developing some algorithm, or we're trying to analyze something which can we explain it to a general environment? Can we apply it to, explain some phenomena that we see in real world? So that's what is kind of where I want to end this. And just to reiterate, I was having a conversation with Zach where he said realistic scenarios lie between perfect randomness and perfect order, and I kind of agree with him. And yeah, I hope I convinced you that network reliability on that graph dynamical systems are very generic tools, and we need to understand which can capture both the structure and the dynamics, and can be applied to a number of different diffusive processes. Thank you again for inviting me, and I can stay on for some questions if time permits.
Thank you so much for your talk. I think the reason, if I may, just for a moment, the reason I was so excited to have you speak with us in particular was this idea that, as origins of life chemists, we tend to either be, working at something like laboratory scale or something we can actually work with and manipulate and understand. Or on the flip side, we work at the planetary scale and we assume, well, everything that's in this box or this, sphere that is our planet is all connected. And eventually, if you give it enough time and you wave your hands enough, everything in that box will eventually, there's a pathway and there will be some chemical pathway open to it. And what I liked about your analysis is that there's this actual way in which we could try to assess if we need compound 1 and compound 2 and compound 3 in some complex network, and they need to be, connected. They need to actually be interacting with one another in real life, unless we've identified one setting where they're all found together. This is a way to estimate the probability through intermediates that they would actually be interacting with one another. Do you see that as being a logical application for this kind of technique, or am I misreading it?
No, I don't know the chemistry aspect, so I'm not going to comment on that one. But I think if we can design, the compounds and interaction, as you said, if we can design it in the way that, we can put it in like a network setting, and we can, it depends on, I would say, how we are defining that experiment or that simulation. Not like lab experiment, but designing this experiment. And if we can set it up in the way that we have our algorithm for network reliability working, then I would be open to this application. And I think it would be great to kind of go beyond and see how it can be used. So I think it would be a fun problem to look at.
Mm-hmm. And have you, do you, beyond the applications that you, just to try to ask, and I think we're going to dive into this in our discussions, I would not place this upon you to try to come up with an answer, but you brought up like the Ising model, and you brought up some of these other applications. Do you foresee any, obvious, potential or obvious kind of problems with applying, say, a technique like this to a chemical system, some based on your knowledge or based on your experience with it?
Could you give maybe an example?
Like, for example, if we're working in a system, for example, you said you're modeling kind of diffusive systems. So
yes.
In this case, do we need to, at the outset, work or build the network description in such a way that we know they're already within potentially diffusive contact with one another?
Yes,
is that an assumption that goes in at the outset, it's baked into your use?
Yes, so one of the things which kind of work with reliability is it doesn't have to be like diffusion at process per se, but as time progresses, and I don't mean time in the sense we assume time, but like, let's say simulation time progresses, right, there is some kind of change in the state. So that's what is required. So the way the reliability framework works is that the probability it calculates is basically, if me as a node, I don't change my state, I'm not really affecting my neighbors who are around me. So there is no change in the interaction. So if the whole probability it calculates is, if I change my state, how does that affect the neighbors? And then how does that affect the other neighbors? And then overall, how does that affect the outcome? So that is what I mean by the diffusive nature or the dynamical nature of the problem. So if I don't change, let's say forever, and nothing changes in the system, then this might not work. But the way it is designed, the framework is designed, is there are some interactions, and it's calculating the change of, how does a change of state kind of affect the process overall. And it doesn't have to be like everybody is changing, but it could be like, that is why I said, like, I start with the assumption that somebody in the network is already infected, and then how that progresses over time. Or it could be that, so I can't start with like a ground state where everybody is relaxed and chilling out. And then like there has to be some kind of perturbation to the system to kind of then see how that translates.
I see. And just as a question of detail, if there aren't any other questions up here, I'm curious You had talked about these expansion series and the order of the expansions. Is there a connection between the number of components in the system and the appropriate order of the expansion?
Yes.
Because I could see, for example, with even expansions, you would have a fundamentally different relationship between x0, x1 versus an odd expansion, and the number of intermediate points that you might have with a larger expansion might lose something, or be adding in a lot of other noise in the middle. Is there a kind of an appropriate relationship between those two?
I don't have a number for you yet, but you're right. Like as the system size increases, it's just a matter of being more accurate. So let's say I have a system of, I don't know, 50 nodes, right? And 50 edges. And I only know like two terms at the beginning and the end. So when I say two terms, that's the first, that is x to the power of 0, that's 1, plus the other term. So that's only 2 term out of, x to the power of 50 possible. So that would affect how accurately my bound is or my interpolation is, but I would still get something to start working with. So that's kind of the approach that we present, like we want to present is like think of, like if you have to calculate the combinatorial using Monte Carlo, it's like sampling a lot of edges to get those tiny ones. So maybe we can use both of them maybe Monte Carlo can help us, solve in the middle part that you just said, because it's quicker to get those samples and get that connection. So maybe we'll get closer and closer to the exact value. So reliability basically is extreme, like it's an NP hard problem to even do an exact estimation. So like what we want to do is get to a more realistic, like let's approximate it with good precision for all practical purposes. So that's what our aim is, and not to get like the most exact solution.
Got it. Wonderful. I think with that, we'll go. I just want to thank you so much for your time, Madhurima, for connecting. And I'm sure we'll be in touch as we explore these kinds of topics in the future. Thank you.
I'll stay on till we have our discussion. But thank you again for inviting me.
Please do. Thank you. We would appreciate that. Our final speaker for this session is Professor Fang Liu at Emory University, and we're excited about Professor Liu's talk because she is going to be exploring, how she has applied these kinds of, network perspectives or the chemical simulation and modeling and dynamic simulations of chemical objects in a network framework. So where you have a lot of different objects interacting with one another under, realistic or modeled chemical rules. And so with that, I'll hand the talk over to Professor Liu. Thank you.
Thanks, Doug, for the introduction. Thank you for inviting me to speak here. So today I'm going to introduce some new computational tools for automated discovery of prebiotic reactions. So the reaction networks are ubiquitous. For example, surface catalysis reaction network exhibit enormous complexity with thousands of species and reactions for even the simplest chemistry. And combustion in engines also involves various types of reaction networks. And the reaction networks in living cells are even more complicated. And finally, prebiotic reactions always involve a large number of reaction networks. So studying the kinetics in reaction networks shed light on the reaction mechanisms, identifying the rate determining steps in the reaction network is crucial for controlling the reactivity and selectivity of chemical processes. So how do we study reaction networks computationally? In all these fields, computational studies are usually very time consuming. So in the conventional approach, we first propose some plausible elementary steps based on experiments, experimental evidence, or chemical intuition. And this involves determining the species and the possible conversions among different species. And then we can do quantum chemistry calculations, such as DFT calculations, to estimate the kinetic parameters with reasonable accuracy, but at a significant computational cost. And finally, we can set up kinetic models based on the reaction rates determined from previously calculated reaction rates or based on existing experimental measurements. And in this long procedure, we have multiple computational challenges. So for the generation of the species, for the prediction of the species involved in the reaction network, there are a lot of uncertainty due to the limited experimental information about the reactions. If each different types of experimental characterization only give us some information about the species. For example, mass spec can tell us the mass of the species, but it cannot distinguish isomers. And IR only gives the information about functional groups in different environments, but we still need to guess the whole structure. And because of that, we need we want to include all possible scenarios. That make a huge network to explore from the computational side. And then for the mechanism, like how one species can transit to another, if we propose the reaction species and steps based on chemical intuition and known reactions, then it's highly possible that we ignore possible important but uncommon reaction pathways. And finally, for the calculation of the reaction rates based on quantum chemistry, these calculations are expensive and often performed in the gas phase to simplify the calculation. But that can deviate from the actual environment where the prebiotic reaction happens. For example, if we want to study prebiotic reactions in the ocean of other planet that has complicated components in the ocean, so then doing the calculation in the gas phase is the default choice for quantum chemistry, but that's not the real environment for the prebiotic reactions to happen. So the various methods have been developed to tackle these challenges. For example, because of the uncertainty of species, many research groups have developed algorithms for automated enumeration of reaction species. And there is a very good review about that. However, our group does not focus on that topic, so I'm not going to go deep in that direction in this talk. So my group focus on several different aspects. So one is like, if once we know the species, there are still many different conformers, and they have this. There are still many different pathways for different conformers, and they can have very different reaction rates. And how can we automatically, efficiently take into consideration of all the conformers? So we developed a Python toolkit, AutoNap, to do that. And for the mechanism, to avoid biased mechanism, we can do automated exploration of reaction mechanism with ab initio molecular dynamics. And to do that, we have developed some tool called nanoreactor. And we also developed the accelerated quantum ab initio molecular dynamics algorithm. So that was some work I did when I was a PhD student. And then to reduce a high computational cost of calculating the reaction rates, we develop acceleration algorithms on the graphical processing unit, and to simulate the real environment of prebiotic reactions, we developed some solvent models and automated workflows. So in this talk, I'm going to introduce these different computational tools to tackle these challenges, highlighted by this the blue fonts. And some tools were developed collaboratively with my lab mates when I was a grad student. Some are developed independently by my group at Emory. So I'm going to introduce each of them. So basically, this slide summarizes what my group focus on right now. So my group is a quantum chemistry and machine learning development group, and we focus on enabling fast and accurate quantum chemistry study. Then to achieve that goal, we need to tackle two major challenges: to accelerate the quantum chemistry methods by developing parallelization algorithms for new hardware, and the other direction is to streamline the workflow. So for the acceleration of the methods, our aim is to resolve the conflict in accuracy and efficiency. So usually for chemistry simulation, there is a conflict in accuracy and efficiency. The more accurate method, like quantum chemistry methods, have very high computational costs that we can only use it to study systems of a few atoms. But in order to study large systems that involves hundreds of thousands of atoms, we usually need to use low-level methods like classical force field. And here we want to enable the description of larger systems with higher level of theory, such that we can explore the prebiotic systems with many atoms, and we can simulate for longer time scale to see what reaction happens. So that is a focus of the development of the TeraChem quantum chemistry package and the nanoreactor. The other direction is to streamline the workflow, because for the setup of the molecular dynamic simulations, it involves many manual operations. So the researcher needs to manually prepare the 3D structure of the molecules, and they need to decide they need to decide how to analyze the calculated results to make them comparable to experimental measurements. And there are also many different theoretical methods we can choose from, and usually we need to determine which method we choose based on our experience. So this whole procedure depend too much on the researcher, and we cannot do it efficiently when we want to study a lot of different systems. So our aim is to streamline this workflow such that we can generate structures efficiently, set up the calculations efficiently, treat the solvent environment efficiently. So for this, we developed some automated workflow that is auto NAB and auto solvent. So I will introduce these different tools one by one. So first, I will start with the ab initio molecular dynamic simulation of prebiotic reaction. So this is some old work I did with my lab mates like in 2014. So here I have a video. I'm going to play the video and introduce what happens for this type of studies. Let me double check that I'm sharing the sound. Okay, I just enable the sound sharing. Hopefully everyone can hear it. So here we are trying to simulate the Euler-Miller reaction, and we started the ab initio molecular dynamic simulation with the following species that includes over 200 atoms, and just set up the ab initio molecular dynamic simulation, let it run by itself, and then we will observe what new molecules are formed in this process. So the species are automatically detected and shown at the bottom of this video. You can see that the species are changing all the time, and the new species formed will be colored, highlighted by these colors. Okay, so we observed the formation of amino acids in the simulation, so that is great. We reproduced the Euler-Miller reaction on the silicon, so in silicon. So then how could we do this? So you might have seen that at the end you can see the time scale for the simulation is like hundreds of picoseconds. So that used to be impossible for ab initio simulation. And that is a key to solve this problem. So we need fast ab initio molecular dynamics. How do we achieve that? We use acceleration techniques to so like first we accelerated quantum chemistry calculation. That is by implementing the formula of the quantum chemistry package, implement that specifically for the graphical processing units. So for this whole simulation was done on the GPUs. We just used a gaming card that people use for video games. And then because the GPUs has a special feature that they are very good for parallel computing. So we need to develop the algorithm to fit the GPU hardware. And with that, we have a much faster ab initio solver. And then in addition to that, we still need to use some techniques to further accelerate these the reactive events, because we hope that there's a great chance for the molecules to meet with each other and to make reaction happen. So we applied some spherical boundary condition, and we periodically push the molecules towards the center, such that we can increase the possibility they meet with each other. And we also simulate this at a very high temperature. Actually, that's not a very good thing, because it does not exactly match what happens for the experiment. But we have to do that to overcome the reaction barriers to make these reactions happen, to observe more reactions. And we were able to simulate like hundreds of picoseconds up to like 1 nanosecond of dynamics per realization. And then in addition to that, we need automated analysis of the species forming during this molecular dynamic simulation. So like what you've seen, the automated detection of the new species, highlight them. And then after that, we will detect the reactions happen for this and do automated refinement of these reactions observed. So like transition state search calculation will be automatically triggered for the reactions detected, and then we can get the kinetic parameters. So then the question is why we must use ab initio molecular dynamics for this. So there are many different level of theories available to perform molecular dynamics. They have very similar ideas. So the idea about running molecular dynamics is that we propagate the Newton equation. So the atoms' positions evolve over time based on the Newton's law. But then the most important thing is that we need to get the forces for the Newton's law. Then how do we get the forces? Then that is a part that is different for different level of theories. So the most commonly used way is the classical force field. For a long time, molecular simulations were only done with classical force field because it's cheap enough, and we can use that to study very big system. Like the protein folding community almost only use that. But however, the traditional classical force field cannot handle like bond breaking and bond formation, because we have to train the force field for each species involved in the simulation. And in contrast, ab initio molecular dynamics is very accurate. It does not assume the existence of any bond. It just look at the structure and you can run the calculation. But then the problem is that ab initio molecular dynamics can be very expensive, even with the relatively low cost theory like DFT. It can be extremely expensive compared to classical force field. But it supports bond formation and bond breaking. So that's why if we want to study reaction network, we want to see chemical reactions to happen, and we need to use the ab initio molecular dynamics. So the following graph compares the classical and the ab initio molecular dynamics. So the bond breaking formation is not available in classical molecular dynamics, but we can run it for a very long time, up to like microseconds, milliseconds, and we can treat systems with up to billions of atoms. And however, the accuracy, like the error can be high. So not the accuracy, it's an energy error can be very high at the order of electron volts. The ab initio molecular dynamics can describe bond formation, bond breaking. It has very low error at kickoff per mole level, but the computational cost can be very high. That's why usually we can only simulate from femtosecond timescale to picosecond, hundreds of picosecond timescale, and even for the hundreds of picosecond timescale, that's only achieved after we do various parallelization acceleration. Then in order to propose bias-free automated mechanism and to automatically explore reaction mechanisms, we need to use the ab initio because we want to see the reactions. But still limited by the computational cost of ab initio molecular dynamics, we can do up to hundreds or thousands of atoms. Sorry for the title, hundreds or thousands of atoms that used to be not available before we developed the faster ab initio calculations. So specifically, the package we work on is TARChem, which is a GPU-based quantum chemistry package. And here we have a review for the development of TARChem over the past 10 years in this review paper published in 2020. All right, and so this figure is just comparing TARChem with CPU-based quantum chemistry code. So the one advantage of TARChem is that we get even more acceleration, more speed ups when we have larger systems to simulate. All right, so that is for the ab initio molecular dynamics. And then next, I'm going to talk about the conformers. So how do we automatically take the different conformers into consideration when we study reaction networks? So these developments were motivated by our collaboration with an experimental group that my collaborators, they did some mass spec study about the polymerized phase synthesis of isoquinoline in the gas phase in tandem mass spec. So the gas phase tandem mass spec is capable of analyzing multiple step reactions. Multiple step reactions can be examined in separate steps by repeated mass selection followed by collision-induced dissociation. So at the end of the day, we can obtain the result in a graph like this. So it shows the abundance of species with different M over Z for different stages of the reaction. However, the M over Z does not distinguish isomers. So we will guess some mechanisms based on the M over Z, and then perform a theoretical calculation to verify the mechanism. And we will repeat this procedure until the proposed mechanism generate product distribution that matches the experimental results. Therefore, we need accurate calculation of the energy profile of this reaction network. However, just calculating this reaction network is not enough, because specifically for mass spec reaction of the isoquinoline, there are many uncertainties about the isomers, which is summarized in the following slide. So for the isoquinoline synthesis reaction related species, there are many mobile protons that can attach to different sites. This forms many isomers, and then we can also have some isomers because of the like the opening and closure of the rings, whether there's a bond or not. And then finally, we can also have different conformers for the same species, like this one. Like these conformers can have very big barrier to convert between different conformers. So that will lead to very different reaction paths. And we should take into consideration of all these possibilities. How do we take all these possibilities into consideration with a automated workflow? So we built AutoNab, which is a Python toolkit that automatically handle all these cases. So for each species in the reaction network, AutoNab automatically generate all the possible conformers. And then for each possible reaction connecting two species, we calculate the energy profile, like the transition state. And then for each energy profile calculation, the essential part is to obtain the activation energy such that we can determine the reaction rate. And these transition state search calculations can be automatically set up. And finally, we can build kinetic models based on these calculations and then determine the product distribution. So for the automated conformer generation, there are already many existing conformer searching tools for organic molecules based on stochastic algorithms and classical force field. But why do we want to develop a separate tool? The motivation is that classical force field usually depends on bond parameters specific to each type of bond, such as carbon-carbon double bond, carbon-hydrogen bond in specific chemical environment. Most existing standard classical force field are parameterized for neutral molecules with paired electrons. So to simulate molecules with charges or molecules with unpaired electrons, such as radicals, there is no existing standard force field. So since the species generated in the mass spec needs to carry charges, so they are not they're not well described by the standard force field. And to get around with this problem, we develop this conformer generator by directly explore all the possible rotations of the dihedrals. But because the conformers generated in this approach are not geometrically optimized, and we cannot efficiently use a standard force field to optimize them, then we want to optimize them at quantum chemistry level of theory. So that is why we want to develop this. And once we generate, so first step, we generate all the possible rotations of these dihedrals, get all the conformations, but then not all the conformers generated by this enumeration is chemically meaningful, because when we generate these, the atoms may clash into each other, and then or they may form bonds that make this molecule a different molecule from the original one we want to study. So we want to get rid of this. So we use the molecular fingerprint to compare the generated conformers with the original species to make sure all the conformers generated are valid. And then after that, we want to further prune the conformers. We want to exclude the very high energy conformers, like the unstable ones with 100 kcal/mol above the most stable conformer. So we exclude those high energy ones. And then surviving conformers are clustered based on the RMSD of the conformer's distance matrix spectrum, such that we want to only look at the featured conformers. And then finally, the lowest energy geometry in each cluster was selected to represent the stable conformer for a particular intermediate. And with this procedure, we can obtain the most representable conformers from the energetic and geometric point of view. And then for each elementary step in the reaction network, we can take into consideration of all the possible reactions between each of the conformer of the reactant and each of the conformer of the product. And then we can set up the transition state search calculation for all these possible combinations. Unfortunately, the transition state calculations are usually time consuming, so it's not practical to search for all those combinations. So rather than doing the combinatorial number of transition state search, we can simplify this procedure by doing a backtracking search. So that means for each conformer, we look at we look at the different for each species. We look at the different conformers and then detect three important reactions. So when we look at this, we look at in a backward approach, starting from the product, look it back, and then we find three representative reactions. One is called the pass L, the lowest barrier pass. The other is the blue pass, the most stable reaction pass. And there may be other reactions, but then we will not take those into consideration. And then so basically we just focus on the pass L and pass S, and then we do this for each of the elementary reaction steps. And then we applied this approach to the isoquinoline reaction, and we detected 27 different routes from the initial reactant to the final product. And then we found the lowest barrier route from the initial reactant to the final product. That is energetically most favorable, and the reaction mechanism actually matches with solution phase isoquinoline synthesis that's already known. So here we're studying the gas phase reaction, but they happen to have the same mechanism. And at the same time, we detected some reaction paths that are not usual, that cannot be found based on existing reaction mechanisms, not chemically intuitive. So we found that's interesting. It may not be, it may not be favorable for this gas phase condition, but it may be important in other conditions. So it's good to detect new reaction mechanisms that's unknown. And then finally, I'm going to talk about the simulation of the solvent environment. So this is achieved by developing the AutoSolve Python toolkit. This is an independent work by my group and Emory. So they are different, mainly two different types of solvent models. The explicit solvent model, where the solvent molecules are explicitly simulated at quantum mechanical or classical mechanical level of theory. So we include those solvent molecules into the simulation. So it gives detailed description of the solvent-solute interaction, but it can be computationally very costly and usually requires high degrees of configuration sampling. We must run molecular dynamics to obtain the different solvation configurations. And on the other side, we have the polarizable continuum models, the implicit solvent models. One of them is polarizable continuum model, but you might have also heard about other types of models like Poisson-Boltzmann model. So the implicit solvent models treats the solvent as a polarizable continuum. So then we treat the solvent effects as a abstract potential applied on the solute. It's more efficient and useful for solvation-free energy evaluation for large molecules. But the downside is that it's a very, very crude description of the solvent. It cannot give accurate description of solvent-solute interaction. And if there is strong interactions like hydrogen bond or reaction between the solvent and solute, then the implicit solvent model cannot be used. However, for the study of reactions, I think the polarizable continuum model is more often used for the quantum chemistry studies because of its convenience. However, for many prebiotic reactions, we want to describe the solvent environment in details. For example, I have a collaboration with Dr. Engelhardt from the University of Minnesota. We want to study the habitability of the ocean of other planets, or like Martian brines, whether Martian brines is good for the stability of DNA RNAs. In that case, we want to describe the solvent mixtures and the ions included in the solvent. So then in that case, polarizable continuum model cannot describe that accurately. So how do we set up calculations for the explicit solvent model efficiently? So they are different approaches to do that. So for like the most traditional approach is to set up the simulation for a large solvent box, run MD simulations to sample the solvation configurations, and then extract the solvation cluster that is small enough for quantum chemistry and run quantum chemistry calculation to evaluate the properties we want to study. But that can be very expensive. So there are other approaches like stochastic generation of the solvent-solute configuration without running the molecular dynamics simulation. However, it's much harder to control the pressure, temperature, etc. for stochastic generation of the solvent-solute configuration without running molecular dynamics. So we still decided to use the molecular dynamic simulations for explicit solvent models, but we want to make this whole procedure more efficient. So to achieve that goal, we developed the automated workflow, AutoSolve, to streamline the explicit solvent calculations. So now we only need the structure of the solute and choose the solvent we want to use, and then our workflow can automatically set up the simulation, including the generation of the solvent box, the force field fitting for the solute, and run the many different stages of the molecular dynamics, and extract the solvated configuration for quantum chemistry calculation. And we have a command line interface and a graphical interface for that. So the graphical interface make it easier to be used by researchers who are not computational chemists. And then we use this on the calculation of redox potential of molecules in the explicit solvent, and now we can efficiently do such calculation for hundreds of molecules. And in the past, usually this type of calculation is only done for a couple of redox systems because of the complexity in setting up this type of simulations. So then in the future, we're going to use this to study the redox potential for like DNA, RNAs in the Martian brine. That tells us whether DNA, RNA are stable in the in the environment of the Martian brine. And finally, I want to quickly mention some recent development in our group related to extreme pressure. So this is related to the development of the solvent models. So we know that the high pressure can significantly change the chemistry. It can induce phase transitions and control reaction rates. And it's not uncommon to see high pressure in planets, like the interior of the planets often have high pressure at the order of gigapascal. And these can be simulated in the lab by some techniques like diamond avocell that can create high pressure on the order of gigapascal up to hundreds of gigapascal. Then how can we do computational chemistry studies that can supplement these experiments under high pressure? So we recently developed the again GPU accelerated algorithm for high pressure, for extreme pressure calculation, and we compared our method with some existing work for argon, and the PV curve agree well with the experiment. And so in the future, we hope that we can also develop some automated workflow for simulating reaction networks under extreme pressure. Then that could be useful for the study of prebiotic reactions under extreme environment, like in the interior of planets. Okay, and then I'd like to thank all of you for your attention. And here is my group and Emery. And I'm ready to take the questions.
Wonderful. Thank you. Thank you, Fang, for that. I didn't realize I had surveyed your work before, and I was familiar with your work with a couple of those tools, but I didn't realize you were so instrumental in developing and applying, all of them. This is fantastic. We did have one question from early in your talk by Junkor Naga. It's in the chat. The Miller-Urey reaction demo is impressive, but I thought that what happened in their experiment was the synthesis of aldehydes and HCN in the gas, followed by amino acid synthesis in the water. So the presence of the two phases appears important. Could you comment on this? I'm presuming the inclusion of multiple phases.
Right. So that work was done purely the ab initio molecular simulation was done purely in the gas phase. So at that time we haven't developed any solvent models. So we didn't take into consideration of the solvent environment. And later, actually, we tried to take that into consideration. And we started with implicit solvent model and tried to add that to the reaction network, to the nanoreactor, to that type of simulation. But we saw that the result is not good with the implicit solvent model. Yeah. So that work was done before any solvent, any solution phase was considered.
Right. If there's anyone else who has questions, feel free to raise your hand or put it in the chat, and we'll pick it up as we go. June thanks you for your response. I was going to ask just a very top level question about the degree to which these tools or different approaches can be tied together, because it seems like we had an actual kind of a related question when we were talking about networks prior to your talk. And the general question was whether, if you're looking at kind of network dynamics on the network level, if you might imagine, another network description for objects at a lower level or at a higher level all being tied together. What are the limits of trying to tie together these kind of multi-level descriptions of complex phenomena might be? And of all the people, I think in our session today, you might be the most poised to answer a question like that, where you're literally working right now to try to get, say an electron field description of an object at one level and all of the dynamics involved in the energy levels at that level, and then trying to connect it to a broader network expansion of, if it's following the rules at this level, what do we expect the behavior to be at another level? Just very generally, could you expand on what you think the difficulties are in trying to capture these multi-level energy flows in a system or in a network?
So I think like one challenge was that how can we, if we think about the multi-level thing, one challenge could be like how we can combine existing knowledge about certain network with this ab initio way of thinking about the reaction network. So like what I was doing, we were actually very focused on the details. So we started from the bottom and tried to go up. But for other researchers, they may have a more top-down knowledge about the reaction networks. So then I think one question is how can we bring these two directions of knowledge together to make a network that can function with both type of knowledge. And actually, I think there are some existing works that can do that. Although that's not for prebiotic chemistry, I think, for example, Todd Martinez's lab at Stanford, so last time I saw some poster presentation from their group. They are trying to bring those two sources of knowledge together to solve combustion reaction networks. And the way to combine them is to use machine learning. So like the machine learning, for example, by using some machine learning model, if the machine learning model, you can inject some top-down knowledge to the machine learning model to tell the model that based on existing domain knowledge, what are the more probable reactions? What are the existing reaction pathway that's already confirmed? And then the machine learning model can then generate some detailed enumeration of possible reaction paths. So unlike what I did for the enumeration of conformer, which has no input knowledge, I just generate everything that's possible, then try to get rid of the less possible ones. Machine learning will have the intelligence to know that what are the ones that it should first generate. And then in that way, we bring the top level knowledge and the detailed enumeration of possible things together. So I think artificial intelligence may be a very promising technology that can solve this problem and bring the different levels of knowledge together.
I see. Wonderful. I think with that, I didn't see any other questions in the chat, but we'll be meeting up for the next for today and tomorrow, and I'm sure that we'll be following up with questions and with potential ways to try to investigate how this might be incorporated with the different activities that our researchers are undertaking. But thank you so much for your time and for your presentation, and make yourself available for this.
Thank you.
With that, I think we'll close out our morning session. We'll take a 1-hour break, and we'll be back at 1 p.m. Eastern with complex systems theory and statistical analysis. So once again, let me thank all of our speakers for session 3 on network theory, and we'll catch everyone back in about 1 hour right here. Thank you.
Yay, that was great.